بدك مساعدة منه شيء؟
هذا اصبروني لا
أبو سمرة
لا أنا بس بدور على طب كيف طلعت محاضرته وهو مش عندي بالتيم شو خلاص أخذ وينه هو
يانس
لا مش موجود
Solomon? Well, we just saw the lecture of this guy, but we can't. He's not here.
No, he's not.
Let me see.
Do you hear me?
Yes. This is the name, right?
Yeah. Yes.
Your Anis.
Co-host. Okay.
Yes. He's what's his name?
Yanis Gerut Anasis.
Hello, Professor Loanis.
Yeah, I'm here.
Okay.
Are you there?
Good.
Yes, do you hear me?
Yes, we can hear you. We'll start in few minutes.
Okay, thank you.
Thank you so much.
So stay on the line, please. Okay? Stay on the line, please. خلينا نروح نقدم من هناك ولا نقدم من هون؟ Huh? Okay, good morning, everybody. Let's start. Our first lecture would be given by Professor Yanis Gerotanisis. I hope I'm pronouncing it correctly. He is at the University of Lunani. Dr. Yanis is at the Department of Chemistry, University of Lunani, Greece. He's also an adjunct professor at the International Center for Chemical and Biological Sciences, University of Karachi. His current research interests include NMR, spectroscopy as applied to bioorganic chemistry, natural products, analytical chemistry, and structural biology. He received his BSc, MS, PhD, and DSc from the University of East Anglia, UK. He has over 180 publications in international journals and 6,800 citations in the international literature. He's also included with the top 2% of scientists according to Stanford University. Stanford University, there's a website for that. He also served as a rector, vice rector of the University of Lunanina, and former vice chairman of the Hellenic Quality Assurance and Accreditation Agency. Dr. Yanis, welcome, and the floor is yours. You have 40 minutes. Thanks. Hello?
Okay, do you hear me?
Yes. Go ahead, please.
Okay, ladies and gentlemen, firstly, I would like to thank the organizers for their kind invitation to make a presentation on ligand observed NMR in natural products and drug research. My presentation will be divided into 3 parts. How to do biotransformations, in other words, how to use the NMR tube.
Prof, can you please put it on presentation mode? Sometimes it refuses, but
Okay, you may proceed, sir.
Is it okay now? Okay, how to use the NMR tube as a bioreactor. The second part will be how to perform and investigate protein natural product interactions. And the last but not least, how to perform NMR experiments at the cellular level. In all those cases, what we observe are only the ligands, nothing else. Why do we need to do chemical transformations or biotransformations of natural products? We do that in order to increase bioavailability and bioactivity of natural products. Quercetin is a typical example. Although there are 5 phenol-type OH groups, its solubility is in the micromolar region. This is contrary to phenol, for example, with solubility which is well above 1 molar in water. What do we have to do, therefore, is to make some conjugations, for example, conjugation with amino acids, which might increase solubility by a factor of more than 25 times. And we hope now, in this way, that we increase the permeability and then the release of the natural product. How can we do those transformations? The classical way, of course, is to perform chemical synthesis, which involves several protection and deprotection steps. It is a time-consuming method, and also it is environmentally rather problematic. The other way is to do enzymatic synthesis with the characteristic properties of high degree of NADO selectivity, mild and green reaction conditions, and in several cases, low operational overheads. Let's take as an example the PCA. Do you see?
Prof, your slide is not moving. Your slide is not moving. We are looking at the overview slide 2. It's the maximum on this. Yes. So the other thing is your slide is not on slide mode. Your presentation is not on presentation mode. Maybe kindly unshare and start resharing, and maybe it will probably work. Okay. Same thing. It's still the same. It's not, just try to put it on presentation mode. Yeah.
Yes,
It's okay. Yeah. It's okay.
So long as it is moving, we can live with this. Yeah.
Is it okay now?
So we're looking at your third slide now. Let us see how it goes. Go ahead, Prof.
Okay, then why do we perform chemical transformation or biotransformations? Certainly in order to increase the bioavailability and bioactivity. Now, quercetin is a typical example with a solubility well below 100 micromolar. And this is contrary, for example, the case of phenol, where the solubility is well over 1 molar in water. So what we need in this case is to make conjugations. For example, a conjugation of a natural product with an amino acid, and this increases solubility by a factor of more than 25 times. So we hope in this way that we increase the permeability and the release of the natural product. How can we perform then those transformations? The classical way is to do a chemical synthesis, which involves several protection and deprotection steps. It is a time-consuming method, and environmentally is rather problematic. Enzymatic synthesis, on the other hand, is characterized by a high degree of NADO selectivity. It involves mild and green reaction conditions, and also usually it has low operational overheads.
So, Prof, sorry, your slide is not moving. It is on the third slide. And maybe I can share my email with you. You can email it and then so that we can share it from here. I don't know.
If you, I don't know if you can do it from there. If you can do it from his side.
Shamshad here. Dear Professor, you just click on share screen, then select first option, screen one, not select PowerPoint.
Okay, okay. Okay.
No, it's fine.
Okay. Let's see.
Super, super. I think
it's even larger screen now.
Yeah, yeah.
Okay, let's take a typical example, the PCA45 dioxygenase. Now the characteristic of this enzyme is that you can achieve the aromatic ring opening, for example, between carbons 4 and 5, and this results in the formation of hemiacetyl group. Now, how can we perform this type of investigation? We just have to make a mixture of the enzyme with a natural product in the NMR tube and follow as a function of time the reaction. Now, what is really characteristic in this case is that we have the prominent peaks of the original natural product, but then after 2 hours, you might notice that there are here some weak signals in the strongly diseased region. After 24 hours, you can see that we have exclusively the reaction product. If we look a little bit more carefully in the strongly diseased region, then you might see that apart from the major peaks, we have also some minor peaks. And this can be attributed to the fact that we have an equilibrium between two forms of the same product. And this is the keto to enol transformation. But how can we predict that indeed we have such an equilibrium? What we have to do is to perform a one-dimensional transient NOE experiment. In other words, you make a selective excitation of this aldehyde proton, and then by performing the NOE experiment, you can see the NOE signal to proton number 5, and this is what you can see here. And this is a negative peak, and this phase is what you expect. On the other hand, however, you might notice that this weak signal here shows a positive signal with respect to the excited signal. This means that indeed we have an exchange processes here. So this is a very useful method, and within few minutes you can follow this type of reactions. Now, another enzyme which is extremely popular is this enzyme here, which has a high degree of NADO selectivity. It also is applicable to a wide range of substrates. It has also a high stability over a wide range of solvents. You can use aqueous solvent, you can use organic solvents, and it shows also high thermal and pH stability. Another property of this enzyme is that it can be physically immobilized on a microporous polymethacrylate resin, and it is known as a novo enzyme. Now, this is sold in the form of some resin beads with diameter of 0.3 to 0.9 millimeter, and it contains approximately 20% weight per enzyme. How can we do now this type of transformation using this immobilized enzyme? We have quercetin, we have vinyl acetate, and the question is, which are the reaction products? The classical method which we use, of course, is to perform the reaction, then to do enzyme filtration, and then to make lyophilization, to perform a variety of fractionations using high pressure liquid chromatography, isolation of the individual products, and then to perform the characterization using a combination of mass spectrometry and MS. But these processes might take several weeks, if not months. Would it be possible to simplify this transformation process? Is it possible, for example, to eliminate the variety of steps and to do just enzyme filtration, take this crude extract and record the proton NMR spectrum? And in this case, what we try to do, for example, is to see especially the OH spectral region. In other words, the spectral region of the phenol type OH groups, because those resonances are well above the aromatic region. But if you record the spectrum, then you will be completely disappointed. The peaks are extremely broad. What can we do in this case is that to arrange a little bit the pH of the solution. Remember that those spectra were recorded in DMSO solution. And if you do that, then you realize that those peaks now become extremely sharp. Of particular interest are those peaks here, which is around 12 ppm. And those peaks are due to the formation of an intramolecular hydrogen bond in this region here. This is a strong 6-member intramolecular hydrogen bond. You can see the formation of 3 very sharp resonances. And one may question, why do we have this extreme sharpening of the peaks? Now, a well-known phenomenon is that if you have labile protons, then the width of the resonances is pH dependent. This can be NH protons, this can be OH protons. And remember also that the vertical axis is a logarithmic scale of the exchange rate and therefore of the line width. Now, this is a typical example. This is the OH region of caffeic acid. You can observe a composite extremely broad peak. But if you arrange the pH, then you get very sharp peaks. One should be quite careful because if you add a large amount of acid, then those resonances increase again. Perhaps the best combination is to use trifluoroacetic acid. So you get extremely sharp resonances in the strongly desilted region, and then you can perform proton-carbon-13 HMBC experiment. And in this case, you can notice, for example, that this proton here shows a variety of carbon-13 cross peaks, which are extremely characteristic for this group of derivatives. And also, you can see here that this proton, in this class of compounds, show a completely different pattern of carbon-13 chemical shift connectivities. So one can go from this proton here to at least 6 different carbons. Then you can go from this proton at least to 6 carbons, some of them in common with the previous one. And then from this proton here, you can make finally the complete, let's say, assignment. Is it possible to even more simplify these processes? Is it possible to mix everything in the NMR tube? The answer is that in principle, yes, you can do that. So you can put now those spherical bits of the immobilized enzyme in the bottom of the NMR tube, and then to hope to follow the reaction products through the diffusion processes. But if you do that, then because you have those solid materials in the bottom of the NMR tube, you can disturb a little bit, let's say, the resolution of the spectrometer. So you have to play around with the amount of the immobilized enzyme which you put. You can play around with the temperature, and you end up with very nice spectra, both in the aromatic region as well as in the OH region. And then you can follow the assignment and also the integration of the reaction products. And you can do, let's say, kinetics. So we can do in situ monitoring, therefore, of multi-product reactions. And our next step is to be able now to immobilize not just enzymes, but also microbes in the bottom of the NMR tube, and then to follow the reaction product. Now, let's move now to the second part of my presentation, how to perform biomolecule natural product interactions. Now, in principle, there are two major methodologies. One is the saturation transfer difference experiment, and the second one is which is a two-dimensional experiment to follow interligand NOEs, and this is known with the acronym in PARMA-NMR. Now, yesterday, it was a nice presentation about this STD-NMR experiment. Let us assume that we have an NMR tube, we have a protein with a concentration in the micromolar region, and we have also two, let's say, natural products with concentration 20 to 100 times more. So you can observe only the resonances of the natural product. Then you make a selective saturation of the protein resonances, and those ligands which interact with the protein, they have, let's say, also selective saturation of the resonances, as you can see here, while the natural product which does not interact with the protein, the resonances remain unaffected. If you subtract the two spectra, you can immediately see the resonances of the ligand which interacts with the protein. Now, serum albumin is a very important protein with a molecular weight of 65 to 66 kilodalton. It has two very important drug sites. Drug site 1, and the typical example is warfarin, and drug site 2, and the typical example is ibuprofen, which is an anti-inflammatory drug. Now, let's assume that we have a natural product, and we would like to see if it shares the same binding site either with warfarin or ibuprofen or both. Now, we can perform then an STD experiment. The first spectrum is the STD spectrum of caprolaic acid in the presence of serum albumin. You can see some very strong STD signals, which means that indeed it interacts. If in the same solution now we add warfarin, then you can see that those resonances are reduced in intensity. In a similar way, if you add ibuprofen, then you can see that those resonances are also decreased in intensity. So one may conclude that caprolaic acid is antagonistic both with warfarin and ibuprofen towards the same binding site. However, this is not the only case. Of course, you may have a competition of both ligands towards the same binding site. In other words, we have a competitive inhibition, but you might have also the so-called allosteric inhibition, where you have another site of the non-competitive inhibitor, which induces a drastic change in the structure, and therefore it eliminates the interaction of the second substrate. How can we make a distinction between those two cases? Now, in this case, we have to use not only the traditional STD, in which case, of course, you make an irradiation of the protein, and then you can see the STD signals of the second ligand. What you can do also is an additional STD NMR experiment where you do an irradiation, not of the protein, but of the first ligand. And in this case, you have a direct magnetization transfer here. You can see now that the intensities, STD intensities, increase relative to the first case. And therefore, one might say, yes, those two ligands are near to each other. Now, this is a very useful method because it is a very simple to perform, and you can get results within 5 to 10 minutes. In our heads, however, another method is much more useful, and this is a two-dimensional NMR experiment. Now, it is well known that if you have a ligand of a small molecular weight, then the NOE is positive and the maximum value is 0.5. If this ligand, however, interacts with a macromolecule, then you have a negative NOE with a magnitude value of -1. Now, let us examine that we have two ligands which share the same binding site. you can transfer magnetization from ligand 1 to the receptor, and then from the receptor, you transform this, let's say, NOE to number 2. As a result now, you will observe interligand NOE connectivities between the two ligands. So you observe not only intra-ligand NOEs, which are the black circles, but also inter-NOEs. Now, let us assume now that those ligands do not share the same common binding site, but binding sites which are near to each other. So you will observe also in this case an NOE, but those NOEs are much more in the number as well as in amplitude. And of course, if you have another case where you have a completely allosteric phenomenon, then in this case, you will not observe any interligand NOEs. Now, let's try now and examine the case whether a natural product, for example, omega-3 free fatty acid, interacts in a competitive way towards ibuprofen in the case of serum albumin. So what you do is that you perform consecutive two-dimensional NOE experiments using 100 millisecond mixing time, 200 milliseconds, 300 milliseconds. And of course, in this case, you might notice very strong red peaks, which are the inter-NOE connectivities. And of course, in this case, one would expect that those two ligands might use the same group for the binding, and this is the deprotonated carboxylic group. And in fact, you can see very strong NOE connectivities between protons 2 and proton 2 and 3 of ibuprofen. However, surprisingly, you will see also connectivities with the terminal groups of ibuprofen. And although the distance here is beyond the potentialities of the NOE experiments. And then we perform docking calculations, and what we found is that in fact within this binding site you have two anchor sites, not one. One anchor site where you have this type of orientation, and there's another one which is exactly the opposite of that. So you have two conformational states. We perform also similar experiments of the omega-3 free fatty acid with warfarin. And again, in this case, you might notice that the red peaks, which are the inter-NOE connectivities, are also very strong. And in this case also, we notice that we have also long-range connectivities. And the result of those experiments is that those interligand NOEs and docking calculations show the presence, even in this case, of two orientations of the free fatty acids in the warfarin binding site. This conformational flexibility is the reason for the inability of the X-ray structure to locate free fatty acids in this binding site. Can we perform also similar experiments, for example, with artemisinin? Can we do also competitive experiments and find out if it binds to warfarin binding site or ibuprofen binding site? In this case, you might notice that there is a proton which has a very characteristic chemical shift. This proton here, which has a chemical shift around 6 ppm because of the presence of two electronegative oxygens. If you add now serum albumin, you might notice a very clear increase in the line width. So artemisinin interacts with HSA. If we perform an STD experiment, we can see a clear STD signal. Now let's perform again competition experiments, two-dimensional NOE experiments. Again, you might see that in black color are the intramolecular NOEs between artemisinin, and the red peaks are the inter-NOEs. And by performing docking calculation, we can find exactly the position of artemisinin within this pocket. And similarly, we can do also other experiments with the other drug. Can we perform those experiments also in mixture analysis? Now, let's perform now a simple proton NMR spectrum of artemisianoa ethyl acetate extract. You might notice that especially in the region between 1 and 2.5 ppm, we have thousands of peaks. Now, let's add in the same solution serum albumin. then you might notice also some broadening of the resonances. Let's perform now an STD experiment. You might notice that this characteristic signal, which is at 6.1 ppm, which very probably is due to H5 proton artemisinin, shows a peak. But again, it's very difficult to make any assignment in this region. So instead of performing only the classical STD experiment, if we can perform the on-resonance STD, you can notice now here an excellent signal and the complete assignment of 4 resonances. So it seems to us that this is a useful experiment which can be performed even in mixture analysis. Now let's move now to the last part of my presentation. Can we perform NMR experiments at a cellular level? Now, you should make a distinction between prokaryotic cells and eukaryotic cells. Now, if we have prokaryotic cells, then in cell NMR samples are directly prepared from isotope-labeled bacterial cultures. If we use, however, eukaryotic cells, then isotope-labeled protein should be first purified from bacterial cultures and then transferred into eukaryotic cells. And this is an extremely difficult process. Is it possible to perform in cell NMR without using isotope-labeled proteins? Now, can we do this type of analysis? Now, a real breakthrough happened in 2005, where they showed that it is possible to make the screening of the interaction of a ligand with unlabeled cell surface receptor. In other words, you might have, for example, integrin, and which is a surface receptor, and then to investigate the interaction with small ligands. And of course, a rather challenging issue is to be able to do those experiments with intramolecular eukaryotic proteins. Now, in this case, again, we have to perform an STD experiment, but those cell suspensions with overexpressed, for example, in transmembrane receptor will be divided into 2 NMR tubes. The first NMR tube contains the cells and the ligand. The second one contains only the cells. We perform for each of them an STD experiment, and then you take the subtraction. The result of this experiment, if you subtract the very strong background from the cells, and then you end up with the signals of the ligand. But how can we know that this ligand interacts with overexpressed transmembrane receptor? We have to repeat the experiment where now the cell suspensions do not contain overexpressed transmembrane. And if those signals are strongly alleviated, then we are certain that this is the case. So we decided to use a similar methodology in order to investigate the interaction of BCL2 proteins, which are anti-apoptotic proteins, and how it is possible to inhibit the reaction of the BCL2 proteins with the use of natural products. Of course, we try to do those experiments utilizing intracellular level NMR. Now, we use exactly the same experiment as before. We can perform STD NMR and also 2-dimensional clear noisy NMR experiments. But the problem here is that the concentration of the BCL2 family proteins is in the low nanomolar concentration. However, there are cases of cancerous cells where this concentration increases by a factor of 5 or 10, but still the concentration is in the nanomolar region. Therefore, whenever we use a ligand, a natural product, as a sensor to investigate the interaction, the relative ratio of the ligand versus BCL2 proteins is over 10,000. Is it possible to have a successful NMR experiment? Now, this is the proton NMR spectrum of eukaryotic BCL2 cells. In other words, these are cells where we have the overexpression of the BCL2 protein. These are the proton NMR spectra of 3 of quercetin and 2 amino acid derivatives. Let's mix now those two at a cellular level. Then you get the spectrum number C. If we look a little bit in the aromatic region, you might see that although we work with cells, the resolution is relatively good. Now let's try now to perform an STD experiment. The STD experiment is successful, but at a cellular level, you have hundreds of proteins. How do we know that we have a specific interaction with the BCL2 protein? Then we used this type of selective inhibitor, intracellular inhibitor of BCL2. And you can see now that the STD signals are strongly alleviated. Can we perform a negative experiment? Let's use taxifolin. Taxifolin has the same structure as quercetin, but there is no double bond between carbons number 2 and 3. Again, we got a very nice spectrum. Then we performed and we got a very successful STD experiment. But when we added this selective inhibitor at a cellular level, you can see that the STD spectrum remains unaffected. So taxifolin does not interact at the cellular level with the protein. Can we perform also another negative experiment? Let's perform a proton NMR spectrum using Euchardt cells, and this is a spectrum, versus pure cells. And you can see now that the signals are strongly alleviated. So can we do now two-dimensional experiments? This is a two-dimensional experiment of the free ligand in buffer solution. You can see NOE connectivities of the opposite sign to that of the diagonal. But at the cellular level, you can observe now that you have the same sign, which means that it interacts, and also you might notice additional NOE connectivities, which seems that the bound ligand relative to the free ligand has a different conformation. However, the problem with the cell NMR is that you need, for sensitivity reasons, high cell densities. And this results in fast depletion of oxygen, cellular nutrients, intracellular pools of ATP. And finally, you have cell apoptosis within, let's say, 2 hours. So within 2 hours, you have to do STD experiment and 2D NOE experiments. And this is not an easy job with regard to two-dimensional NOE experiment. A real breakthrough happened 2 years ago when they used Aragos gel in order to keep and avoid the precipitation of cells. And this, it is then combined with this accessory, which is now commercially available for flow in and flow out. And in this case, the scientists succeeded to have cell viability for over 72 hours. Within 72 hours, you can do not only two-dimensional NOE experiment, but you can follow also the evolution of the natural products as a function of time, how that different analytes are transformed to others at the cellular level. So NMR is a useful method. Even you can use an NMR tube as a bioreactor. You can do very useful experiments with regard to protein-natural product interactions. And last but not least, you can do also some useful in-cell NMR experiments looking just of the ligands, not on the proteins. Finally, I would like to thank my colleagues and especially Professor Giakos Andreas, a genius collaborator, and Dr. Alexandra Primikiri, who performed the in-cell NMR experiments. Thank you very much. Do you hear me?
Yes, sir, we can hear you.
All right.
Kindly share screen again.
Do you hear me now?
Yes.
Yeah.
Okay, well, thank you again for a very informative lecture. And on behalf of the organizers, I would like to thank you so much for contributing to this workshop. We hope to see you in our country in person.
Thank you very much. Sorry for all those difficulties in the connection.
Okay, thanks much. Yeah. You're gonna use this one. Then I'll go there. All right.
No problem. Time problem. So the ones online, can you see the slides? Hello?
No, sir, we can see only black screen. Stop sharing and select screen.
Can you go to settings of sound?
Okay. No video. Okay.
Settings.
Hello, can you hear us? The ones online, please confirm.
Yes, we can see.
Hello?
Yes, Dr.
Okay,
Suleiman.
can hear. Good. Super.
Yes, we can see, but not in slideshow mode. Kindly change your slide.
Yes,
Can they hear you?
So he wants to introduce you first. Where is it? No, it's not coming on. Yeah, I think it's trying.
So now I have the full pleasure to start another session and to listen to another state-of-the-art research from a world-class scientist, Professor Christian Petzel from the Institute of Biochemistry and Molecular Biology, Hamburg, Germany, who's going to talk to us today about latest development in serial crystallography and its application in structure-based drug discovery. Actually, I have a very prestigious CV for Professor Petzel. He studied physics at the University of Göttingen and finished his dissertation, doctoral dissertation in Berlin, 1986, Department of Chemistry, Institute of Crystallography. He held a lot of positions in EMBL in Europe, Hamburg, in the University of Berlin, Department of Chemistry, and served as a lecturer for biophysics and scientific staff member in University Hospital of Hamburg, Eppendorf. Served also as a professor of University of Hamburg in 2000, and a doctor of science. He got the Doctor of Science from Academy of Science in Sofia, Bulgaria, Institute of Organic Chemistry 2002, And since 2004, he served as a professor in the University of Hamburg. Since 2015, he served as a professor in the International University of Sao Paulo in Brazil. He's a member and advisory board member in many scientific societies all over the world. And his key aspects of activity, he's going to talk to us today about the structural infection biology, crystallogenesis, structure and dynamics of nucleic acids, specific plant lectins, and genome proteomics. Please share me with welcoming Professor Pesdemdas.
Yeah, dear Chairman, thank you for the kind introduction. Dear organizers, thank you for inviting me and taking care. And it's my pleasure to join this very interesting workshop. And I know I'm a little bit of satellite. I'm working in X-ray crystallography. The community is here with high expertise in NMR and mass spectroscopy. On display, so you see already the title I structured. I will start with an introduction, new and upcoming methods in X-ray structure analysis. So we're using crystals to look at atomic resolution in terms of drug discovery in the field of infection biology. This is in North Germany a main topic. And then I will bring some examples targeting antibiotic resistance and corona research. In the background picture, you see already one, my colleague here introduced yesterday, coronavirus research. This is a peer approval, the papain-like protease. We investigated structure in complex with natural compounds we see from ICCBS in Karachi. And but more details will follow. It's not stuck. It's on.
This should be okay now.
Okay. I can use this one.
Yeah.
Thank you. A short comment here because there was some discussion yesterday. So the workshop is on spectroscopy. So I define spectroscopy as a technique and method. Diffraction is a physical phenomenon. So I'm teacher. I'm just bringing this comment in here. And whoops. Now
It's a pink.
I'm kicked out.
Don't worry, no, I think
It's behind. I hope it's stable now and moving forward. Okay, so
sorry.
this.
No, this thing should go away because it's blocking your.
It's in the right one.
That's it looks different.
Minimize.
Yeah. Okay.
Somewhere here. Representing.
Okay.
Okay, we are coming close now. So I'm located in Hamburg, North Germany. So it's a nice city. You see here we have a lake inside the city, a harbor here also. This was my flight distance. So I heard from Professor Zitisch, he was 32 hours. So the distance here is only 7 hours from Hamburg to reach flying time to Amman. And oops, really? Okay, but Hamburg is also known for photon science. So we have the German synchrotron facility here since the 1960s of the last century. So we have storage rings producing ultra brilliant X-ray radiation. Here the harbor is behind here. You see the river Elbe. And this facility is also called Petra 3. I know Petra you have in Jordan. This is an historical and very important city here. Petra is the abbreviation, is German complicated word, Positron-Elektron-Tandem-Ringanlage. So here, positrons or electrons traveling with the speed of light and producing this very intensive X-ray radiation. I will come in the tutorials back to this topic. And here underground, we have 3.1 kilometers underground, the European free electron laser, which is on top. You see here the intensity, Konrad Röntgen, 1897 X-ray tubes, the first synchrotrons coming up, and free electron lasers. Here's the intensity, produce almost the photon power, which is bringing the sun on half of the Earth in square centimeters. So it's a tremendous intensity used for material science research and structural biology. Here we have the scheme. I used one picture from a textbook from Bernard Rowe. So it's a clear one-way road. So you produce your protein.
Sorry, Prof. The online people, they are not seeing your screen.
Not seeing your screen.
Yeah, there is not yet. Sorry. I think.
I have to go back.
Yeah, yes. This one. If I begin. Yeah.
Activate the video.
No, he didn't want to. Yeah, so where is your PowerPoint? So I don't know. This one here, this one. Yeah. Okay, this one.
No, next.
This one.
Yeah. It's hidden behind.
Is he co-host?
Yeah.
Yeah. Yes.
As a co-host?
Yeah. Yeah.
Okay.
Yeah.
Let's go back to this one. Now, I hope the participants online can see this slide. So I just summarize X-ray crystal analysis here. So you produce your protein, but can be any biomolecule. You can crystallize it. So there are lots of methods known here, and I have some crystals with me. They can be tiny in the micrometer range. And then you use X-rays, you expose the crystals. Today you have very advanced electronic detectors. You collect the diffraction pattern and you see the structure at atomic resolution. And this is used, here you see a structure of an enzyme and a unit cell in the crystal. So the active site is shown in a type of cartoon presentation. You can use crystals to soak compounds in crystals and to analyze the structure at atomic resolution, or you can co-crystallize. I come back to this. This is again was one of my questions, or Professor Wahab was targeting this solubility of compounds is certainly an issue. And we follow the rule of 5. If you're in compound or screening or lead discovery, normally you start with small compounds, less than 500 Daltons. They have to offer some H bonds. They have to accept H bonds. They should be water, ethanol, or DMSO solubility, and they should have some flexibility. I'm still in the introduction. I'm showing this slide. This is showing the first crystal structure by Kendo and Holmes and co-workers here. At this time, they were collecting data on paper strips. I remember this time. This was when I started my diploma thesis. This was just going out after you had magnetic tapes, and today you have everything on a small USB stick. Single crystal here, flash frozen, X-rays, data will be collected and analyzed, and you calculate your electron density. And here you have again one crystal sitting in a loop, and you have, in most cases, a liquid nitrogen stream to cool the crystals to avoid radiation damage. This is a historical, let's say, I call it conventional method. valid for many years and still in use, of course, in most places. Upcoming along the last 5 years, where with this very intensive X-ray sources, you can collect data in the second and millisecond range. This is tremendous. When I did my PhD thesis, I was in days and weeks collecting data. Now we are in the second range, and means you can collect data also down to the millisecond range. You can follow dynamics in protein and biomolecular structures. So we spray crystal suspensions on a tape. They are X-rayed, and here you have the data collection, or you have liquid jets. You spray crystals, passing in vacuum, passing and X-ray. And upcoming now is this ship technology. You have ships, they can have up to several thousand of these little wells. You can just pipette a suspension of crystals on these ships and collect your data. Of course, here you need only one crystal, maybe a little bigger. Here you need 10 to 6 to 10 to 9 crystals. But technology in crystallization is these days very advanced. If you know once a condition to produce a big crystal, you can just manipulate the condition to obtain many small crystals. And this is showing one beamline at Petra 3. So the X-rays are coming from this direction. Here's the place where the crystal will be aligned. You use normally these days robots. Robots have the advantage they are working day and night. They are not drinking tea or coffee. And humans are sometimes too slow. So if you have your crystals here in this liquid system, they're collecting data 24/7. And I will show later, you can send crystals from any place of the world. It's a small, I have one here in my pocket. So there's, it's coming out here. So you have several hundred of crystals in this little thing. You can post into a synchrotron, and you can follow the data collection from your home or from your institute. And I have to press this button again. This was here for frozen crystals, and here we have ships. They're sitting in a hotel. They have several hundred crystals on top. So this robot is bringing the crystals in position, and then you can collect the data remote. And we offer workshops every year. Last year in summer, we had a nice workshop, international. So we have people from all continents, also from Pakistan. They are just trained in preparing crystals and using the remote procedures. And now I'm showing a slide of the protein data bank. This is for me the home of protein structures. You can obtain coordinates for soil structures, but also this web link is providing all software you need to analyze structures. And the good news is everything international on the same level. So we use the same tools wherever you are, you use the same software wherever you are to look at structures, to solve structures. And I'm close to our medical department, so we have medical doctors, they're crystallizing and solving structures. They don't want to know the physics behind. They're just using this, pressing the button, go ahead, and to obtain structural information. And as you see, just was looking yesterday, so the protein data bank has now more than 200,000 experimentally solved structures. And X-ray crystal, here the methods, X-ray crystallography is still the working house. Of course, you have NMR, and upcoming is also cryo-EM. And I remember the end of 21, AlphaFold came up, was in structural biology a super surprise. Some colleagues were under shock because AlphaFold is capable to predict structures with a very high precision. So we thought at this time we are all unemployed, now in the world. It's a tremendous tool to predict structures. And there are also workshops now, webinars. Students will be trained in AlphaFold. So it's a game changer in structural biology and structural prediction. It's tremendous, and I'm super impressed. We are also using, actually, AlphaFold is using 2 lines. It looks in genetic databases and structure-based databases and linking all the information, mainly from deposited structures in the PDP, to predict the 3-dimensional structures. So almost overnight, all human, or all in our body, all human living, let's say, protein structures were solved. That was the reason why we had this peak. Maybe I go back one slide here. You see that several 200,000 structures came overnight towards the protein data bank. However, as I mentioned, it's a very nice program system. The precision now is in the angstrom regime. And I can mention already here, for drug discovery, you need higher precision. So drug discovery and lead discovery is remaining a field for experimental science. So remaining limitations are you can predict only single chain protein structures, but we know many proteins are oligomeric, dimeric, tetrameric, and hexameric. So you need to perform experiments. Disordered and unstructured regions are difficult to predict. Here we have X-ray small angle scattering, no dynamics, and for lead and drug discovery, of course, you need to do the experiment. And just for the students here and the PhD and master students, regularly the AlphaFold is offering webinars. You can link in, you register from any place you are and join one of the seminars and you learn all tools. So the future of X-ray crystallography is, of course, drug discovery and molecular time domains. I mentioned before, today we can collect data in the millisecond regime, and this is where life is active. So enzyme reactions are in the millisecond regime. If you look from a physical point of view, or if you ask physics, the milliseconds are super slow. They want to have picoseconds, femtoseconds, but for life science, millisecond experiments are good. And what you're seeing here is one who is penicillin binding protein, where most of the, say, antibiotics are going towards or they're targeting this type of enzyme producing this cell membrane. And you see there's a lot of dynamics. Coming back to this topic, also Linus Paulik and Daniel Koshland already predicted that the key lock principle known very well in structure-based drug discovery is not a solid. So the block is moving and the key need to be flexible. Just showing again, this was the first time resolved movie almost 120 years ago and was supported by the Stanford, later founder of the Stanford University. He was a railway baron, we say, and he hired several photographers to make this first time resolved movie, developing special film and special cameras. In between, one photographer who was in prison because he killed his wife, but he came out later, so there are lots of funny, not funny, but some history behind. I'm coming to this because this is our research field now. We are using these tools for data collection, of course, single crystal diffraction remains, to catch up time resolved data. And here's a scheme of a synchrotron radiation facility. You have several beam lines, and you have intensities in this range. We have third and fourth generation. So they are super intensive. You have many experiments at the same time. But on top, the free electron lasers coming up. I will explain later the principle here and the principle here, which is slightly different to X-ray tube, providing tremendous intensity. So you can collect data also down to the picosecond regime. And just to say, or to mention, X-rays, you cannot see, you cannot smell, but you see here, this is from a synchrotron, is melting a copper block. So they have tremendous intensity these days. Of course, medical doctors, they use dentists, they use, of course, X-ray sources also. They have good intensity, but they are not as intensive as synchrotron sources today. And when the free electron lasers were designed, almost the first experiments in this direction were done 20 years ago, part of the community was saying this radiation is too strong, everything will explode. And however, the development went ahead and the principle came up, diffraction before destruction. So there's enough, this is a protein hit by free electron laser beam, but has enough time to send a signal for data collection. So we have a bunch of synchrotron radiation facilities all over the world. Upcoming are free electron lasers, also located almost on every continent, and are rephased again, so you can be at any place. You need only to post or send your samples. And I know Sesame in the region is coming up, which was before in Berlin, was a previous synchrotron facility in Berlin. I was checking the homepage. So they say X-ray crystallography is under construction. So I hope it will be finalized. But I guess the experts here in the community know more about. This is a view on the free electron laser we have in Hamburg. Hamburg is a federal state. It's just crossing the border to the next federal state, which is very clever in terms of funding. So 2 federal states. So it's 400 meters behind the border, underground. So you see here the lab building, but you have to go 3 floors down to reach the experimental area, which is here. So it looks, this is real countryside. We have cows here around, but the radiation is like a subway tunnel passing through and reaching this area where we have 5, 6 different experiments, one in life science, the others are in material science, spectroscopy, and so on. Now changing towards one of my scientific targets I want to present now. So the Hamburg metropolitan area, we have many institutions that are focusing on infection biology, microbiology, structural biology, up to medical hospitals analyzing resistance effects of bacteria. And we are, of course, in drug discovery. So we are looking at structures. So it's like to find lead compounds. You know, this picture is looking like find a needle in a haystack. And I was screening also yesterday, also the Jordanian community, actually all the world you see here, color-coded world map, is facing the increasing resistance, antibiotic resistance, which is really emergent, and it needs and recovers active research activities to identify new antibiotic compounds. There are also articles saying now this is correlated in the last 3 years also with the corona pandemic. And one of our targets, together with medical doctors and scientists in microbiology, we are targeting the methicillin-resistant Staphylococcus aureus, which is known to cause also hospital infections, which is very Staphylococcus aureus and epidermis are the most unfriendly bacteria known today, and there are only a few drugs known as last-line antibiotics, which you can use to target this type of infection. So they're colonized in the nasal area, but the infection part, if they reach lung or other organs, patients are in a very unfortunate situation these days. And this is a type of textbook figure. So antibiotic resistance mechanisms are known. So bacteria fighting against antibiotics or limiting uptake, modifying the drug, inactivating the drug is the most important target in drug discovery now, and active drug efflux. And here are the enzymes we are working on also, of course, international, many scientists are in the field, targeting the lactamase, which is hydrolyzing the penicillin-like antibiotics. And just to remember, I guess the community here in the auditorium knows, if you start from the beginning, from scratch, you can, it needs sometimes more than 15 years from the idea passing the first experiments, phase 1, phase 3, to bring a drug or a new drug on the market. Upcoming in the last 3-4 years, also pushed by the corona pandemic, was a drug repurposing approaches. So if you find a drug which is already in application to treat another disease, you are very lucky because you can skip all this time-consuming, not only time-consuming, but also money-consuming parts here in drug discovery. And for drug repurposing, you may need 3 to 5 years only. And along the last years, there were also, you see new databases, drug repurposing hub, the searching tools. If you plan an experiment in this direction, you can go to one of these web pages, and there's a lot of information stored. You can define your enzyme you want to look at, and so on and so on. So this is one, the drug repurposing hub, and another one is a drug bank. So you can screen for compounds and their deposition of several thousand compounds and the targets, the enzymes, it's mostly enzymes they are targeting. Our research field here, one is we are targeting a beta-lactamase of Staphylococcus aureus and Staphylococcus epidermis, and is called CTX-M14, which is known today. So we isolated, not we, but microbiology also from patients. We sequenced and prepared plasmid for the large-scale protein production, and to look at high resolution and also in space. So our units are Angstroms, but also to see time resolve. What is the enzyme doing, if it's targeting and if it's binding, for example, an antibiotic, which are shown here, avibactam, these are the known as last line antibiotics now in applications, particularly in hospitals. What is the enzyme really doing? So how it is hydrolyzing such drugs. And along our literature research some years ago, we identified that boronate-based compounds, which are shown here on this side, there's a boronic acid, coming back to be of interest. They are known, for example, in biotechnology. If you produce enzymes, for example, for washing powder, you want to store the enzymes happily for a few weeks and months. They're producing, or they're adding a little amount of boronic acid, which is inhibiting the enzyme. As soon as I stay with washing powder, if you bring the washing powder in solution, the boronic acid is diffusing out and the enzyme is active. If the enzyme is not inhibited, it would hydrolyze themselves before you are applying the washing machine. And this principle that boronic acid can also inhibit other proteases and enzymes, the lactamases, serine protease, came up to be very interesting. So the first active inhibitors were developed and are in application. And we started a repurposing approach. We were looking at other inhibitors of enzymes, serine proteases, having, say, boronic warhead. And one is the ixazomib, known as anti-cancer drug. This is a very latest anti-cancer drug inhibiting the proteasome. And we could solve the structure. Just showing again the beta-lactamase. Here we have a penicillin-like structure, which is going to target the penicillin binding protein. However, this drug is hydrolyzed by the beta-lactamase. This is the target. The beta-lactamase is protecting the penicillin binding enzyme, hydrolyzing the penicillin core. And we could co-crystallize, and we are happy. We had high resolution, so we see the structure at 1.3 angstrom. We see here the electron density and the active site serine here, which is in this location, and a covalent bond onto the boronic side of the bortezomib. And this was very interesting, or is very interesting. In principle, you can go for repurposing using this type of drug also to treat infections, bacterial infections. However, we know that bortezomib and ixazomib are also last-line cancer drugs, so they have a lot of side effects. So our idea is to understand more and maybe to modify together with pharmacists this type of inhibitor. Here we have ixazomib, which is the latest anti-cancer drug, proteasome inhibitor. We see reasonable IC50 values, but knowing again it has tremendous side effects. At this time, you would not apply to treat infections in the first order. Now I come back to our X-ray crystallography approaches. So we're using tape drive, you spray crystals to collect data at different time points in the millisecond region, liquid jet and the ship technology. I prefer this one because the material you need to do this type of experiments are reasonably low and is very effective. But all three methods, you can collect time-resolved data in the millisecond range at different time points. I'm starting one movie because here's some mystery. This is the enzyme active site, and it's known to bind ions in the native conformation. So we see sulfate ions and here, which has two conformations, and we were adding boronic acid to see what's going on in the active site. Till now, you have only static pictures, native or inhibited, but this is a very important, it's not moving anymore, okay, process. If you add a substrate or an inhibitor, this ion has to move. So in this case, we added just boronic acid, and we see it slowly kicking out the sulfate, bringing it to a new disordered site, and we measure the volume. And it's not water. So normally you consider that you have solvent water bound. And again, I come back to the discussion yesterday with Professor Wahab. She also prefers experiments. This type of things you cannot predict by molecular dynamics or modeling. So this was one of the first experiments we did, and this is a summary. So we see first 100% occupancy, different time, and here we see a reorientation of the sulfate. We see less occupancy, and we see that the electron density is building up at the active serine, the O gamma, and forming finally a soft type of covalent conformation here. So this was a type of pilot experiment, and I have to sum up. In the next type of experiment, we found out that glycerol and boronic acid together. This is a movie showing how we know boronic acid is binding, kicking out, or kicking by sides the ion, the sulfate ion. And if you add glycerol, it forms a type of ester bond in the active site. So very interesting. You see the buildup of the formation of the electron density. We see, I'm of course giving the interpretation here, the formation of an ester bond covalently bound to the serine. And for this, we collected several hundred data sets. What is going up in a few minutes, you have these data sets. And now we understand in which way the other boron-based inhibitors targeting beta-lactamases are binding. And this is a nice summary here. So you see the beginning, pre-soaked. You see a cyclic ester is forming with, if you add glycerol, and this bonds, you see the electron density increasing over time, and you see a nice bond formation. So this will help future drug discovery investigation, because you have for the first time time-resolved information about bond formation. Many other groups looking like targeting bond break or hydrolytic events. In this case, we follow bond formation. And this you can do with time-resolved crystallography. This was just briefly pinpointing this topic. I'm coming now to our SARS-CoV-2 investigations and what we did in the past. This I collected also yesterday from the dashboard of WHO. And of course, we are happy that mostly unfortunate events of corona pandemic are over. Of course, we have to take care. And you see here the number of deaths till now. And WHO is monitoring also new occurrence or daily reported infections. So we are still have corona, and corona will, of course, probably stay forever. And as mentioned by you also, we have to consider future drug discovery investigations. This is just a short summary. We have seen 2 presentations in the last days. So we are happy that we have very effective vaccines these days. So they suppress the virus entry here by, how to say, interacting with the spike protein. The development of efficient drugs is still, I say, in a pilot situation. There are some investigations well published. We have the first drugs from Pfizer, but they are not well received. This is Palaxovir. It has a high concentration of 2, say, infectors or inhibitors, and it has a number of side effects also to be mentioned. So when Corona was coming up early 2020, we started to have a research collaboration on the DAISY campus. So we collected 100 scientists from the area of Hamburg with different expertise, and our goal was to target the main protease and the papain-like protease. This should be a K, not line. And on the main protease, there were already lots of investigations on coronavirus 1 and other associated virus done before. So you can produce, crystallize, and apply for co-crystallization and soaking experiments. The papain-like protease, also PL-Pro, is more unfriendly. So we could start with the main protease almost after 3 weeks. So we were working day and night in protein production and crystallization. The PL-Pro is nasty. So you can express, but it's not folded. So there are lots of issues in between to obtain the folded protein in a crystal form. And this is a consortium. We had the DAISY Institute, 2 universities, one Fraunhofer having a drug libraries available in the area. So we had this consortium, about 100 researchers are involved. And the second approach we obtained on short notice also from Karachi, ICCBS. Thanks again for this natural compounds to screen the PL-Pro. And this is a summary of work, maybe also a tutorial for the students here. You have your plasmid, you do protein expression, purification, and crystallization. You first screen to obtain crystallization conditions over many conditions. If you have good or identified ones, you go ahead, produce in bulk amount crystals, and you need to harvest crystals, shock freeze. You have here the robot, and robot is working day and night. I mentioned you do the data collection here, and then you look at the structures. This was also as one student, she was for PhD in Hamburg, Hina Anderlet from the Multan University in Pakistan. She was working very hard in the harvesting harvesting crystals. At this time, we had a lot of restrictions. So most of the university was closed, so we had to consider special precaution procedures. And again, here the crystallization facility, the robot. And this is running 1 minute movie. I hope it's starting. Yeah, here. Showing the whole procedure. And we had scientists at this time from Moscow, we had from Cameroon, experts in crystallography, collecting data. So we were sharing the data collection. The robot was bringing, and scientists were assigned for 2 and 3 hours to collect data and process data, even from Sao Paulo, Campinas, US. We had 20 every day doing the remote data collection. Here you see the crystal. The robot is bringing to the place, centering, and then the data collection is starting. I'll push a little bit because so you press buttons if you are confident, and you see here white spots are the diffraction pattern, and also DAISY was supporting tremendously. All this remote business was not really established at this time, to say thanks to Corona in this case. So all these procedures were prepared and assigned. Here we see the main protease. So we had 2,500 data sets. In color, the first compounds were assigned for binding. In total, we identified something like 49 compounds, and one was a real hit. So in parallel, we started cell assays and activity assays to see the inhibition rate. And we could publish this in the journal Science. So I was one of the coordinators, and in yellow underlined colleagues from Pakistan joining this enterprise. And I just briefly summarize, this was one approach going on. The new publication is coming up. So we had also, okay, 5 minutes, that's good. So we are targeting the papain-like protease, which is very interesting, difficult to handle. And I come back to you also, this is known to as a multifunctional, it's an enzyme, but also modulating the how to say, activities of the host, of the human. So it is modifying ESG15 is known to be involved in the cytokine pathway here. So the enzyme is deubiquitinating this type of molecular ESG15, suppressing the immune reaction and supporting the excessive inflammatory reaction. So we know that patients infected by corona, the immune system is going down, and unfortunately is going down. And the PA-Pro is one of the main players just facilitating this. As mentioned before, we received the compounds. We had some custom issues. That's if you have international collaborations, you need to consider custom and transport and paperwork. And we obtained the compounds. We started the same procedure, co-crystallization of the compounds, and we looked at structures. And interestingly, we found 3 compounds binding at the site here. This is the active site. There's some flexibility here, but this is the site where the ESG15 molecule is binding. So we are inhibiting the binding of ESG15, means the deubiquitination. All 3 compounds look from a distance somehow a little bit similar. They're also repurposing drugs. They are known to be used to treat heart failures. They are antioxidants out here. So they have already some application. This is very interesting. And here are the compounds overlaid. So they're interestingly binding almost in the same region and inhibiting the enzyme activity. Here you see the electron densities of the 3 compounds, the interactions, and we were investigating further. We prepared this type of plots to see these are H bonds and ionic interactions. So the compounds are somehow different, and also the interactions are somehow different. And of course, we are looking in parallel at other coronavirus PLL2s, because corona was one event. Maybe the next virus is coming in the near future. Understood that the bird fly virus is approaching South America, maybe coming soon to Europe. So of course you have to keep your eye open to see if there is some homology between other coronavirus, say in this case, papain-like protease. And just briefly, so this activity assay here, 100%. If you use these compounds, yeah, of course, not inhibiting 100% water solubility is one point, but again, green tea seems to be the overall good thing you have to take every day several times. So this shows the inhibition rate of the compounds. Of course, we did other investigation, IC50 we measured, so we see some effect. And of course, the mission is you're not taking one drug to inhibit one enzyme. So we identified for the main protease, so maybe a cocktail of drugs targeting 2 vital enzymes of the virus, maybe a good combination to cure corona in future. And we're coming back to red wine. So this is one compound here, is known to be in red wine and olive oil. And this is still now our favorite here. It has tremendous activity, and we initiated also BSL-3 laboratory experiments, applying a special mice system, a social mount, and Vero cells. And we have very interesting results, and they're coming out in one of the next Nature Biology journals. So it's interesting, but now, of course, this was one of the preliminary publications we assigned to your birthday last September when I was in Karachi. I was presenting this book. And because of the natural compounds, this is a unique facility. And acknowledgement, my colleagues at DAISY and XFEL, the free electron laser, is a tremendous biolab we are using. And this is my group. So we are only 3 Germans here, 1, 2, 3. There's guys from Scotland, the others are quite international, and you can recognize some PhD students from Pakistan. Before I hand over to a tutorial, I can encourage again. So if you can produce crystals in any lab, wherever you are, You can harvest all these tools are international standard. You can prepare in small boxes and transport to any synchrotron or X-ray facility, because now they don't have users. You send your sample, they do the remote data collection, you join by Zoom, you save travel money, and it's very effective. And thank you for.
It's very excellent, world-class research and very resourceful as well presentation for all of us for continuous learning and continuous training. And now the floor is open for one or two questions all of the time. Please.
Thank you, Prof. Very interesting presentation. In your screening, you were using the protease, the main protease, and also the copies. And I've seen a lot of work being done using the RNA dependent RNA, which I think is one of the main enzymes responsible for viral replication. In my work, I focus on two. One is on the division of the S protein, for the virus, and the ACE2 for the host. This one in the entry, and then on the that's what you did on the main protease, and also the RNA dependent. Because I think that if you just based on one, in your case is two, patient also, you may miss the activity of the compound you spray on the division of the entry and the binding, and also RNA polymerase. Because I've seen papers, most of them focusing on the RNA polymerase activity, although of course some of them I think on the main or treat. Can you comment?
No, that's absolutely true. So we decided when we started this to form this Corona research collaboration to focus on the enzymes, because we all have experience in enzyme technology, production and crystallization. There's also the E protein, which is also an interesting target. And as you say, the polymerase. And we had contact with Pfizer. So Pfizer started something similar, but I learned that Pfizer has 300 scientists starting on this topic, and they have a good budget. So we are just starting, and all of us were not in viral research, but we had to read to learn all these things. But at the end was very effective, receiving compounds, natural compounds, also drug repurposing. And we have a collaboration via my association in Sao Paulo with the BSL-3 lab. They started immediately. You know how long it takes. This ethical approval, you need to have mice which are happy under low pressure. So I was myself many times in the lab preparing all these experiments. And PeerPro, also if you look in the protein data bank, there's one section only for coronavirus structures. You see the polymerase, you see the main protease, and the PeerPro. The E protein is coming up. And so you have to be open-minded, but to be clear. Also, I would like to mention here, crystallography will not catch up all conditions because you co-crystallize. So the compound may change the crystallization conditions. There's no guarantee. So I'm in favor to combine mass spectroscopy and NMR crystallography because you see some structures, but certainly you cannot see more structures. This is the power of NMR technology. As you work in solution, you may see different complexes. So you have to combine methods as possible in your area. This is a message to the students to keep the eye open, to see what is available to do some, for example, thesis research activities to be complementary.
This one, how does AlphaFold compare with Meta AI? Do you have any idea the software or the big confirmation agents?
Today, I think AlphaFold is the favorite. They call it AlphaFold 2. It has limitations, so you cannot see complexes. but maybe AlphaFold 3 and 4, I'm sure there are coming more versions. I'm really impressed about the performance. And you have a free license you can use from your home lab. And it's very interesting to use.
So how different is it from homology modeling? AlphaFold. Does not depend on experimental.
No, it looks in a genetic data bank and mainly in the protein data bank catching up coordinates. And it has a precision in the range of 1 angstrom, but not considering solvent ions and other issues till now. And you can see only monomers till now. So there are many proteins coming up in oligomeric. The main protease, we have trimers, hexamers, so oligomers till now, or complexes, protein RNA cannot be predicted. But maybe in the future there certainly.
I invite all of you to, if you have further questions, discuss it with Professor Benjamin during the coffee break. Now, please join me in thanking Professor Benjamin. Thank you very much.
According to the schedule, I think we have a practical session mainly for students. So this will be led by.
No, right, practical session.
Oh. So this will be as a practical session.
Can be extended for also another 10 minutes.
Demo material.
Do you need the mic or? Very good. Very good.
I have one PowerPoint for this. I'm showing it.
So you share it again. So that's so good for that. Also have
Yeah. Even this one, I can put it back. Sorry.
Okay, now it's my pleasure to present a short tutorial on X-ray crystallography, the protein crystallography using the method to look at biomolecules. Of course, no doubt we are filling one summer and winter course, and I'm compressing a little bit, means I will pinpoint the most essential sections and parts. Here, 1/6 scarf. So here you see the yeah, again, this is a summary of the procedure I showed before. You express, you purify your body, you crystallize, and then you have only one formula utilized to measure the electron density here in rho at each position XYZ in the unit cells. And here's one formula which has one common parameter which is on the back of your conference book, which is common to NMR spectroscopy. And we have intensities and phases. And anyway, this is the only formula you put calculator intensity from experimental data. Here, experimental data, these are these dots here, and this requires Braggs law. Use my little pointer. Let's see if they're working. Also see if you have ordered material like this, see spots. So if you use a laser pointer, you go to a shop and test the silk or some other material, but I'm using a laser pointer between 500 nanometers, and of course here the crystal, if you have crystal, you use X-ray. Just pointing out, if you have monochromatic radiation and exposing, in this case, the color structure from intensity, sorry, if you have intensity. From intensity you see on the wall, you can calculate the structure of the silk stuff. So the same principle we are using in X-ray crystallography, but instead of laser pointer, we are using X-rays, and because X-ray radiation is bringing you the passive wavelengths in the range of the atomic bond. This is just the principle behind, and here again, crystals, they can be tiny. They can be invisible for the human eye, so because we have very intensive radiation, you produce via Bragg's law, coming to this diffraction pattern, and this morning in the car we discussed in textbooks, you find the detector was film, X-ray film, and maybe you can show around. For more than 80 years, it was a good detector up to the 90s of the last century, and then the first electronic detectors came up. And because if you use film, you need the dark room, you need the film, you need to convert the black spots to intensities, to white. There's a lot of work behind. Today, it is super fast. However, many structures were solved applying X-ray film. Then you have phasing. Here's the part, is theory, and you obtain your model. Why using X-rays? Again, this is for the students here. So we can use light microscopes. They are very advanced. Now you find articles talking about super resolution in cell biology. You can see many things, but light has some limitations. You know which type of limitations, or up to which resolution you can look at structures approximately. There's one simple formula in cell biology. It's half of the wavelengths. This is the maximum resolution. Means you can see particles up to 200 nanometers. So light is good, but unfortunately, it would be nice if you can see proteins with a light microscope you cannot see. Now we have electrons. You can also accelerate electron microscopy. This is one point, but electrons are difficult to handle, and why you're not using electrons, you can accelerate electrons themselves also to have very low wavelengths. The reason is they're very unfriendly to material, so they will burn our sample. And since 1912, Lauer detected the first diffraction pattern, so we are using X-rays. It's the most easy and cheap to produce. There are very good home sources these days, and the X-ray, the crystals, the type of a lens, we produce this pattern. Here's Bragg's law. I'm coming back to this. So X-rays are the most suitable way to look at molecules in material science, organic chemistry, life science. Here are neutrons. Neutrons are also used, and this was one question yesterday also by Professor Schultz. You can use neutrons. What is the advantage and disadvantage? Disadvantages, they are difficult to produce, expensive, you need reactors. And however, they behave the same. And the second question is X-rays interacting with electrons. So they're scattered by electrons, whatever material it is. And the advantage in life science, I can distribute here, we have the 20 amino acids here. So whole life is built up only from 20 amino acids. The amino acid chain. And coming back to Professor Schwartz, so we have carbon, nitrogen, oxygen. So they all have almost the same number of electrons. So they're almost scattering in the same way. So what you see at the end is the electron density, just a second here, is a smooth distribution of this type of atoms. What we are not seeing are H atoms, because they have only one electron. However, in life science, if you know the other positions, if you want to see their programs, they can predict the H atom. So every protein, for example, when it has, we say, 2,000 non-H atoms, it has 2,000 H atoms on top. So actually, you don't want to see them at the beginning because it's confusing. If you want to see particular cases, you use neutrons because they are scattered at the core of them. So this is complementary, and in particular cases, if you want to see an enzyme reaction, you can apply for neutron reactor types. In Europe, we have 2 or 3 reactors only for this type of experiments. Okay, so just a summary, light microscope has limitations, would be nice to see, but it's magnification and X-ray diffraction. So the crystal is a type of a lens, scale like this. And the crystal has the molecules are sitting ordered, and then Bragg's law is working, and we can process the data. So one of the key formulas, I guess my colleague is also talking on this later, is Bragg's law. Coming back, the X-ray source was first developed by Konrad Röntgen in Würzburg in 1897. And however, he didn't know what it is. So he was X-raying his own hand, you know, the famous figure with the ring finger, and he named it X-rays because X, for him, was not clear what it is. And Lauer came, first diffraction pattern he was interpreting was in 1912, and Bragg, actually it was father and son, William and Henry Bragg was a team. So they understood, they understood finally what's going on. So you have 2 waves. This is ordered material, can be any type of material, organic material or life science. You have 2 waves crossing this material, and when the difference between the paths of the wave is 1 full or multiple full of the wavelengths, they are forming interference. So the spot is the individual spots on the detector referring to a layer of layers. Of course, this is in the textbook, you find very simple drawings. But the Bragg's law is valid in this case, for example, for 2,000 different positions. But to keep it easy for us humans, in textbooks, the wave is coming here and going out here. In reality, of course, the radiation is horizontal. Just again, why X-rays here? So you see wavelengths in nanometers here. We have X-rays here after gamma radiation coming, which is more powerful. Physicists, they name also, they use kilo electron volts to define the energy. But X-rays are perfect because we are coming in the range of the chemical bond. You know, chemical bond here in this case is roughly 1.5, 1.4 angstrom, and X-rays are exactly 1.5 wavelengths. So it's fitting perfect together, easy to produce, one formula, you collect your data, and you see your electron density in all dimensions, and you can, of course, you need some brain later, you need to model the electron density, you have to bring your amino acids, in this case, or nucleic acids or compounds in. And at the end, you have coordinates, X, Y, Z, in a unit cell with a very high precision. And again, just to remember you, these are the amino acids. What is the difference in amino acids? It's also simple. We have only 20 amino acids. If you go to Toyota, Mercedes, or Audi and say, if you 20 compounds, or please build a car, they would laugh. So, however, life is constructed by only 20 amino acids, and the difference are physics. They are small and big, they are polar and nonpolar. So evolution was very clever. And of course, from the single chain, which is going around, I guess, and the chain is magnified with plastic model. 1 centimeter is 1 angstrom. So we have a factor of 10 to 10 magnification. Of course, you have secondary structure, and from the secondary structure, you have domains, and at the end, you have the quasi structure. And we know we have several millions of hundreds of thousands of different complexes in life science and chemistry. So this was the first structure, sodium chloride. Then we had urease in 26, pepsin later on. The first structure, myoglobin, cancer parents, received the Nobel Prize in history. And just showing again, at this time, there were no laptops or computer graphics. They used wood models to show the electron density. Here's a data set on paper strips. And chemists and biochemists were very surprised because up to this time, they did not consider secondary structure. So many biochemists were under shock seeing this type of sausage stuff. However, Linus Pauling were one predicting this, and Alejandra and others. They were communicating by post. At this time, there was no fax or email. And you find textbooks showing this type of correspondence. They were the only scientists predicting that the peptide chain cannot be random, it needs to be ordered. And later on, we had computer graphics. So these are the first wire state. This was metal, copper metal. When I started my diploma thesis, I was obliged to work with this type of models, the first 3D structures. And here's a Nobel Prize event. Rosalind Franklin analyzed the DNA structure. Very unfortunately, she passed early. It's interesting to mention, up to this time, it was not clear that X-rays are dangerous, cell imaging. So that many experiments were done in an open room. So you can expect if you collect data for weeks, then you of course collect unfortunate radiation events. In the late 90s or early 90s of the last century, the first 3D computers came up. I remember we had 3 in Germany, 1 in Munich, 1 in Hamburg, and 1 in Berlin by Mercedes, so they designed cars. However, we were allowed to use these computers part-time before we had to go for a training course. But now every laptop you have very advanced software. Here we have a training course for master students. We have also Beamer. They can go produce stereo images, and you can walk to a protein. You go to the active site and do your modeling. And all the steps are today very advanced. So you are not, even if you are unexperienced, the software is helping you. It knows all 20 amino acids. It is warning you if you do mistakes and alert you. And as you know, many of the important structures were published in high impact factor journals. I come to a second point here, almost in the abstract or even in the title of the publication, you find the information to which resolution the structure was solved. This is important to keep in mind. As higher the resolution is, as higher the precision is. And for drug discovery, a topic in this conference here is also drug discovery. You need the resolution in the range of 2.5 or lower, means higher than the H-bond. H-bond distance is 2.7 to 3.1 angstrom. You need to have this resolution to make some clear interpretation. If you have only a resolution of 3.5 angstrom, you can model everything, looks nice, but you have to be careful for the interpretation. Here's 4.5, which is low, but it's a huge complex. So you need enzymes, most probably small molecules, provide higher resolution. So this is again for the students in the audience, the radar criteria defining the resolution is most important. So this, you can resolve this just briefly, but these points are too close, you cannot separate. And you see here electron density, I can read this here, you have 3 angstrom, the phenylalanine 2.5 angstrom and 1.2. So the resolution, the electron density shown is the most important. Again, so this we have to keep very proper in mind. And we define resolution as at this resolution you can identify side chains. Here you can identify solvent and ions, and here is going toward high resolution. In the protein data bank, I call the mother of the data bank, there's also statistics. And these are showing that all structures in the protein data bank deposited have some good resolution up to 2 angstrom, indicating that crystallization, crystal quality, and X-ray sources are emerging very good. So most of the data you are seeing are in good quality. Again, here I'm stressing this very much. You see medium, low resolution, and here high resolution. Here are the 20 amino acids again, and now I'll briefly pinpoint crystallization, so nobody should be afraid, because you can say if a biomolecule is, or how to proceed. If your boss is giving you some material and say, please make crystals in 3 days or 4 days. So what is necessary? So point 1 is you check purity. So protein, I stay with protein, can be also RNA, needs to be pure. It needs to be folded. You have to check CD or apply CD spectroscopy. And we are checking with light scattering. You have a cuvette, dynamic light scattering. The homogeneity, in this case, we have 1 radius. And the system is collecting overnight data. If you see this, there's no excuse, it should crystallize. If you have all these parameters well monitored, you are very safe that you can obtain crystals. And light scattering you can do in 30 seconds, you have this information, and you can further use. Now, just pinpoint on crystallization methods. So most textbooks you find this type of phase diagram to adjust the precipitant and protein concentration. And if all parameters meet well, you are maybe here in crystalline nucleation zone, crystals are growing. But you are of course blind. Also most proteins are colorless. So to obtain information where crystals are growing, you use this type of plates in the laboratory. One is circulating. And you screen many conditions along protein concentration and precipitant concentration. And in this plate, you have your precipitant here, you have your protein solution here on a maybe cover glass or the plane are distributed sitting. And you mix the protein and 50-50 with the precipitant solution. Means your precipitant has higher concentration here compared to the droplet. And taking slowly, as you see in this figure, water from the protein droplet. Means the concentration is slowly growing. Important is slow, it has to be smooth, and then forcing to be in the nucleation range, and then you see crystals. And we have a bunch of different methods. There are also textbooks, there are international conferences on this topic, because this is the only bottleneck. If you have crystals, all other procedures are easy, I would say, and clear. However, the crystallization is a real issue, and just showing some textbooks and okay. I'm starting one movie here. So we have one case, so PhD student in my group, I hope you can see contrast. Crystals are growing in 90 seconds, and many small ones. It is a huge complex. So it's pure, homogeneous, and well folded. And of course, this is some unique example. Mostly you need 3, 4, 5 days to obtain crystals, but this was we are very happy. We monitored this experiment also by light scattering. You see starting material, here's the radius, and later on crystals. One other method is counter diffusion. You can do it also easily. You have a capillary. You bring on one side your precipitant, on the other side the protein solution. You close it properly, and then you have counter diffusion. So you're also passing the phase diagram, and then if your conditions are well, or you can see crystals, and I have shown a few examples here. The small rockets we can go on.
I prepared a simple experiment, and nice here is a type of field solution you can take in your pocket. Of course, plates you into that horizontal, so you can prepare such an experiment and send to X-ray source or do at home or whatever. Just show them easy. And this crystals are big, so they looks good today. You can use smaller crystals. That's a very handy method. So if you have once the conditions approximate, you can modify and use also other procedures. So this is the one just going around. And of course, in laboratories, you have robots and other facilities. Here we have one robot in our lab, which is preparing in 20 seconds, 96 conditions. So students, they can do pipetting, but you need a protocol, and after 1 year, maybe you cannot read the protocol. Today you have e-protocols, but if you use a robot, it's producing also a nice book. So they have a bunch of procedures in automation. And let's go to the summary. So here I have one slide showing different methods. All methods have advantages and disadvantages. They need some procedure, but there's a bulk of information on today. So as I mentioned before, I have some colleagues in the clinic, even the medical doctor started to crystallize in their home lab, and they're happy if they obtain crystals. After conventional, you freeze is the one standard method to protect from radiation damage. And again, all the tools you are using today, they're uniform international. You can utilize and join training courses. You can fish, bring in nitrogen, freeze, and send in a duo. These are procedures we are also training master students first with lysozyme and then go ahead. Just coming to the last section of the tutorial. If you see crystals, you're of course happy because you know the protein is in order in the crystal. So Bragg's law as well. Just to remember, it's not a matchbox or like some humans lining up. So they have distinct orientations, and there are a number of orientations known. I've shown this. So there's symmetry between the molecules you are seeing. And again, don't worry, there are 230 different symmetries are known today. You find in textbooks, but software is also knowing this. I stay with my example, medical doctors. They don't want to know this. They press return. They're bypassing these sections. But if you're in a training course, you want to know what's behind. So you can ignore this or you can understand. And in life science, we don't have mirror symmetry. The chain is passing through the room, is showing the amino acids, and we know they are all in L conformation. We know we have D peptides, but in the protein, we have only one conformation, the L conformation. For nucleic acids, you have defuranosis. So it means it's reducing to 65 space groups known for biomolecular crystallography. X-ray sources is Konrad Röntgen behind. He designed this type of tube. He pictures his own hand. He didn't know what is really going on, so he named it X-rays at this time. And however, today we have very advanced X-ray sources. I know ICCBS has also one very advanced unit, so it's very handy to use. You can do most of the experiments. Now, important just to see that the radiation is monochromatic like my pointer. So the X-rays we are using are from conventional source are 1.4821 angstrom. So this is important. Then you obtain, you cannot use polychromatic radiation. Again, Bragg, they identified the principle behind, it's called Bragg's law. So that the wave difference, the length difference is need to be one or multiple of the wavelengths. And this was a breakthrough. Again, X-ray, conventional X-ray sources, copper block electrons, they are very advanced and convenient today. We have also one in my lab. I'm 200 meters close to the synchrotron, but for many cases we are using conventional radiation. Synchrotrons came up in the late 70s of the last century. And I remember also one comment from Professor Chaudhary yesterday. The physics at this time were not happy when they detected it was discovered 1947 at General Electric, because it was a waste of energy. They were super worried. So electrons traveling, this is a model, this is one of the latest synchrotrons in Chernobyl. They're losing energy and you have to pump energy back. I give you one example. DESY in Hamburg is using electricity on its own compared to a village of 50,000 people. So electricity bill is tremendous. However, this was used first for high energy physics, colliding positons and electrons, and now each continent or country has one or two synchrotrons. The principle is you produce these particles, you accelerate, here's a pre-accelerator, and they're moving with the speed, almost speed of light. So they're moving very fast, and at this speed, they're producing radiation. It's named synchrotron. Why? Because the particles need to be focused via magnets. Also here we are using superconducting magnets. However, the magnetic field is traveling synchronous with the particles. Otherwise, the electricity bill would be too much. So that's the reason why this radiation has the name synchrotron radiation. And it's very useful these days. So here's the conventional source, synchrotrons. And you see here the magnitudes of intensity gain and free electron lasers. And this is in Japan. Just to mention, this synchrotron radiation is much more focused than this point. The radiation is going in focus of nanometers. So it means the ground where the synchrotron is constructed need to be very stable. So in Japan has similar situations as we recently heard about Turkey. So when Earth is moving, earthquakes. So they went on one mountain and cut the tip of a mountain, which was mostly level. In Hamburg, we are afraid of subway. So the DESY is always measuring the vibrations of the, how to say, concrete plate to forward stable radiation. And yeah. Again, here the beam is coming. Here's our sample, and here's the electronic detector. Now approaching the end, there's one situation, just to briefly mention. Here's the formula, electron density. Here's your experimental intensity, F, they call structure factor. And here's the phase, because we know electromagnetic radiation has intensity and a phase, 0 to 360 degrees. We can only measure the intensity. So the phase angle need to be calculated on top. Just to show, sometimes you see textbooks with the wave character and the intensity. So this situation, we don't know the phase of the intensity. This is called also the phase problem, but today I'm not going in detail here. We can solve this. We have three ways, direct methods. We don't need to consider this for small molecules. And we have an experimental area, it's a little bit more efforts, and molecular. Again, don't worry, this is very covered by software, need to be considered. However, the solving of the phase problem from Bragg was taking almost 30 years. And why it was so difficult at this time now to solve, you see importance of the phase angle here. You have intensity and phase angle. Intensity, phase angle. And Karl and Hauptmann, two scientists, prepared many models. They used the diffraction of a duck. Say like that, we can use a duck. I use the silk squared. And you take the phase information from the duck and the intensity from a cat. Then you obtain an image of a duck. So just highlighting the phase information was more important than the intensities you are measuring. So you need to take care about phasing. However, this would be one complete lecture. And as you see from the data bank information, this situation is nicely solved. And at the end, okay, last slide here. You obtain in three-dimensional electron density. You sit in front of a computer, can be laptop, you model your amino acids, you obtain a final structure, and then you have this is experimental data, intensities, phase information, and Fourier transform. We have common with NMR spectroscopy, electron density. You model your amino acids, and then you obtain coordinates in angstrom units with the precision of 3 digits behind the comma. Here you see a threonine, proline. So for each non-hydrogen atom, you find this kind of information in the file you normally deposit with the protein data bank. And it's like this for master students and PhDs as part of the thesis. If you submit, you need to submit before sending a manuscript to a journal. The journals want to know the called PDP code. Each protein has a code in the protein data bank, and the protein data bank is checking this very precisely. They're checking covalent bonds, non-covalent bonds from a Hundert criteria. It's like a technical check. And if you have the PDP code, then your theses are safe because this is the last quality check. And you can send your manuscript because the journals want to see also the PDP code, then they're knowing the structure is well solved. It was very compressed, so maybe I stop here. Again, we have tutorials and courses going all in these details, and all the young people don't need to worry. This is easy to roll this. And again, I emphasize crystallizations.
So much for this very interesting presentation and training. Please join me again in thank Professor President effort. Questions or comments of sorts, please during the coffee break. We have a lot of time, 20 minutes. Please be here before 11:30. Thank you very much.
Professor,
I'm sorry for the time.
Yeah, yes, sure.
You mentioned you did a neutron experiment for just this is general for protein crystals.
So thank you very much, our online participant. We're going for a tea break. We'll be back at 11:30 in about how many minutes? In 20 minutes. Shukran.
Neutron reflection data for 2 enzymes.
بس تنتين او تنتين يعني في راسه بعدين بيجي السامع على الماس بيجي سامع على الماس لا السامع هذا شيء زيادة فدوى المفروض كان فدوى بعد هذا لما يجي فراس بعد فراس فدوى هون فدوى بتيجي بعدين يجي فراس بعد الغداء بعد الغداء بعد السيشن لا بعد السيشن بعد فدوى معنا هذا تيجي بعد فدوى فراس بتيجي مكان بعد فدوى من Hello. Okay, after our next lecture after the coffee break is called solid state nuclear magnetic resonance spectroscopy, will be given by okay, أنا بقدمك بعدين خلاص. Okay, yes, Anna. عشان احكي عنك كثير شوي. Okay, apparently there is a change in schedule. So our next speaker, which is part of the training session, right, is Professor Sameh Abu Zaid. So Sameh Abu Zaid, he was introduced yesterday. So I'm not going to repeat what was said yesterday. You see him, he's a very nice, handsome young man. And he will talk about the concept and application of mass spectroscopy for small molecules, which will be part of a training session for students, right? Okay, Mr. Sameh. أيوة يهزم
So thank you very much for the introduction. First of all, we will give for maybe 10 minutes or 15 minutes a very brief introduction to this technique. And please, if you are an expert really in any of the spectroscopic technique, I invite you to leave at once this whole. we will be very basic and begin from scratch actually, and give a very brief and detailed introduction to the technique of mass spectrometry, targeting students, graduate students who are really new to this field. So first, I'd like to give a brief introduction to what is mass spectrometry, and let's call it here spectrometry, not a spectroscopy. We will deal with another technique other than that we listened today and yesterday, not very much interaction of the light or interaction of electromagnetic waves with the matter, with our molecules, with our compounds, but a different methodology. So we call it mass spectrometry, and we will define it as, an instrumental method used for analysis to identify compounds according to what is known as MZ, or mass to charge ratio. Mass to charge ratio. And also abundance of these masses in the gas phase ions. So first of all, from this slide, we can know that we are dealing here not with neutral molecules, but we are dealing with ions. This technique, mass spectrometry, doesn't see neutral molecules, only deal with ions, as we will see in the next slides. The basic principle of mass spectrometry, as I said, it's like we will begin from the very beginning, from scratch actually to this technology, is a charged particle. When it moves in a magnetic field, the pathway of this particle gets bent or deflected, as you can see in this photo. So this is a charged particle, and when it meets a magnetic field here, its pathway gets bent, and this bend or this deflection, bending in the pathway depends on the mass to charge, depends on how much heavier the ion, because heavier particle will be less deflected, and also more charged particle will be higher deflected. So what we use here is known as mass spectrometer, and we have several processes which occurs inside this mass spectrometer. The instrument we use for this technique, first of all, we have to make ionization. As I said, here, this technology or this technique only deals with ions, doesn't deal, it's blind for the neutral molecules. So after we make ionization in the gas state, we want to sort or analyze or separate these ions, and then we want to detect these ions and send our data from the detector to an data handling system like computer to give us the output, which in the form of mass spectrum. So these are the basic process inside the mass spectrometer, the basic processes which happened in this technique in mass spectrometry. Ionization, please remember, and analysis of the ion or sorting of the ions, and then detection of these ions to give us the mass spectrum. But we have to know that two important things here. All of these processes occur mainly in most of the instruments in the gas phase. So usually the compound or we analyze has to be volatile. And also we need vacuum to prevent ions from colliding with each other. We need to see the ions in the detector as separate ions. So this gas phase condition and this vacuum condition will help us to get ions away from each other, and then we can see them neat in the mass spectrum, and then we can analyze our compounds. We can know what is our compound. And this needs actually some of the prerequisite for having this technology working in a proper way. When I say vacuum here, we have to maintain vacuum in our instruments. So before we decide to go for this technology, to buy this technology, we have to prepare our lab for a good infrastructure from the electricity point of view to have a stable and continuous power supply, because if we cut the electricity, we have to re-establish the vacuum conditions, and this will complicate the process for us. So a real take-home message for all of us here is before we are thinking about purchasing an instrument like this, maybe over $1 million US, if it's high-tech instrument, we have to maintain or we have to establish a good infrastructure for the electricity supply to work in a proper way thereafter. Then let's have a look on what is a mass spectrometer is. If we have an sample here and introduced into what is known as an ionizer, the sample M will be ionized, will form ions, and will be attracted by electricity or bi-electrical. This is the ion source where compounds are getting ionized by different technique, as we will talk later, and then enter into this slit to go in this pathway, and its path will get bent, as you can see, like this. But some of the ions will go like this, some of the ions go this in orange pathway, and some will go in the green pathway, depending on their molecular weight. This is known as the analyzer of the mass integral part of mass spectrum. Sorts the ion according to their masses and their charges by using several electric and magnetic principles. Then finally, what ions will reach the detector is what ions we will document here, or we will see here in our output, which is the mass spectrum. So let's watch this very simple movie, actually, which can show this process. So, please, from the
Okay,
from
then
the flash memory and then share. Stop sharing this one. Flash memory. No, it's not here.
no, I will. I think if I do this, I can easily go to the flash. Go here.
Animations. Maybe it doesn't have software.
No, it's just not responding.
Okay, no problem. We can continue.
Okay, I think we continue with the
What's the point?
yeah.
Concept and application.
Oh,
Again.
this is just refusing to PowerPoint, not responding.
So when maybe the movie is not for technical purposes not working, but anyway, to be available with you in the electronic material, you can scan it from here, from your card. We'll go back to our PowerPoint presentation.
You can send us the movie later on.
Yes, it's very simple. Yes. So our output here is what is known as what we'll get out from the instrument is a mass spectrum. And if you look at this mass spectrum, you can find x-axis and y-axis. On the x-axis, it's mass per charge, and on the y-axis, the relative intensity or relative abundance of these masses. So as you can see, there is one signal or one peak reaching the 100. We call this base peak or base ion peak. And you can find other peaks for the molecule, which we can analyze, as we will see later, and understand or try to get the molecule structure or molecular structure for our compound. Sharing.
I think this should be okay.
Two important pieces of information here is we have the highest ion the highest peak here, which we call the base peak, and the last peak here we can see, which represents usually for most of the techniques we use in this mass spectrometry, the molecular ion peak, showing the molecular weight of our compound. So we get the molecular weight, and we got also fragments which will help us to know the structure of our compound. So let's go back. As I said, there are two main processes which occur in the mass spectrometer, inside the instrument of mass spectrometer. The ionization, getting ions, as we said, mass doesn't know or is blind towards the neutral molecules. So we first have to make ionization, and we make ionization actually by several techniques, as you can see here. Some of these techniques are hard. We call it like electron impact ionization, and we usually use this for molecules which can be volatilized. And then we discovered that this technique doesn't give us the molecular ion. When it's first invented long time ago, we discovered that we need another soft ionization techniques to make it visible for us, the molecular ion visible for us, so we can expect the empirical formula or molecular formula for our compound, not only getting fragments, not only fragmenting the molecules. So other techniques has been developed for ionization, like chemical ionization, like fast atom bombardment ionization, usually called CI or FAB mass spectrometry, like electrospray ionization, abbreviated as ESI mass spectrometry, and usually used in research articles for a very high molecular weight compounds, and multi or matrix assistant laser desorption ionization technique. These techniques actually can be used for several compounds. As you can see in this graph, we have in the x-axis the polarity of our compounds or the compounds we need to analyze, and on the y-axis the molecular weight of the compound. So if we can look at this technique, usually used in the gas chromatography mass spectrometry instruments, the EI mass, it's used for relatively very non-polar compounds and low molecular weight compounds. If we can go up here, other technique like atmospheric pressure chemical ionization or atmospheric pressure photoionization, or the ESI electrospray ionization used for the highly polar and for high molecular weight compound. So this is another take-home message for all of us today. Before I decide which instrument I have to use or have to send my sample, or which technique I have to use, or which instrument I have to buy or purchase, I have to know what is my application is used for. Am I going to analyze non-polar compounds or highly polar compounds? Am I going to analyze low molecular weight or high molecular weight like proteins and peptides? Let's go quickly on this. Yes. To go into the training, these are showing this slide showing you the advantage and disadvantage of electron ionization. You can see it later, and this is the schematic representation for EI mass spectrometer, and this is what we get from the EI mass spectrometer. Then compare it with other techniques, the ESI, we use a much less electron volt or much less energy, not to fragment. As I said, this is softer ionization technique, and this is the schematic representation for the ESI mass ionizer, how it works. We get output from the LC device, we evaporate the solvent, and this particle, which contain more than one molecule, charged, can be exposed or take a fission to get the single ions here, which can be analyzed later. So this is usually used in either positive or negative mode to analyze high molecular weight compounds, very suitable for proteins, for tannins, for other macromolecules. This slide showing you the difference between EI mass and ESI mass. For example, in the output here, we can see a lot of fragments in EI mass, while in the ESI mass, we can just see usually the molecular ion of our analyte. The other techniques like chemical ionization, atmospheric pressure, chemical ionization, FAB mass and matrix multi or matrix assistant laser desorption ionization also can be used for different purposes. The most important thing, we choose the right technique for our application or for our case we are going to deal with. So next is the other very important step in mass spectrometry, the sorting of ions we get from the ionizer. We use different electrical and magnetic principles for sorting these ions. I want to separate these ions either by quadrupole or triple quadrupole, or what is known as time of flight or orbital trap. Different companies giving different technologies here. For example, the quadrupole, we have 4 quadrupoles connected to electrical signal. And this is the triple quad or QQQ, in which we get our ions. We select one of these ions, we make another fragmentation in a collision cell in the intermediate step, and then the fragment ions are selected again. That's why we call it QQQ or triple quad ionization technique. Sorry for this very long introduction. Actually, now we go for the practical training. I give you a very simple cases. We can use what we have just said in this very simple analysis. One mass spectrometry connected to gas chromatography, and the other one mass spectrometry connected to LC or liquid chromatography from our work in our group. Practical training.
No, yes, this one, isn't it?
Yes.
Yeah. What's the beginning? So, sorry.
So as I said, we have this slide. If you'd like to see it on your computer or your mobile, you can just scan the QR code in your card here, or you can follow with me on this on the screen. First, we'll use mass spectrometry to analyze essential oil obtained from plants. Many plants are called aromatic plants. They have some aroma, so they contain what is known as essential oil, very related compounds, simple molecules, usually non-polar. We need to analyze what are these compounds and what are the quantities of these compounds. Mass spectrometry coupled to a technique called gas chromatography is a very useful in this case. We obtain our volatile oil or essential oil by a very simple device here using hydrodistillation. Maybe most of us know this technology to get our oil even in a very low amount and analyze it using the technique called GC mass or gas chromatography coupled with mass spectrometry to give a chart like this. This is the chromatographic separation or gas chromatographic separation for the essential oil. It can give you some of these, some signals like this, maybe 30 or 40 or more signals, just separated molecules depending on their abundance. This is the highest abundant compound. Very low molecules here, very low compounds here. So the x-axis again, this is the time. The compounds are eluted from gas chromatography machine, and this is their abundance as they reach the detector. But we never know from this chart, from gas chromatography, what are these compounds are. We need to identify this compound, except of course we have an authentic for each of these compound, and we get the same retention time. It is eluted at the same retention time, and even this is not conclusive to say this compound is so for its chemical structure. So we need to answer this question together. Why does the x-axis labeled relative abundance? What's referred to, as I said, this is the abundance of the compounds as they are released to the detector. Which signal have the highest relative abundance here? If we go back to the chromatogram, we can find the signal at 20 minutes, 20.31 minutes. This is the highest signal, and depending on this high signal, which reaches the 100, we will calculate the abundance of the other signals in the chart. So we will consider this a chromatographic signal at 20.3 minutes. I give you a hint, this corresponding to one molecule, a kind of monoterpenoid molecule, 10 carbon compound molecule called citronellol. What happened for citronellol to create a detector response in the GC? Citronellol here is giving a signal by carbonization or by being ignited to give a signal like this, depending on its amount in the oil we got from the plant. So it gives us the highest signal or the highest peak. So how can we identify these compounds? We have different methods. Either we use what is known as retention index. We inject our sample along with other compounds called normal alkanes, starting from C8 to C18, under identical, the same experimental conditions, and then we compare what we get here, retention index, to the reference book called Adams in identification, very long, very old book, and we can get retention index. And the retention index here is very specific to our compound, whatever instrument we used, whatever the conditions we have used. Or we can go for each peak we got in the gas chromatography, and we get the mass spectrum of this compound and compare it with the online available libraries like NIST or Willy library, as we would see just in the later slides. And then we calculate the retention times, also another method. If we have a authentic compound or standard for these compounds, we can create just the retention time, not the retention index of these compounds. For example, the signal at 20.31 minutes, we get the mass spectrum for this signal. We can see highest signal here and some other fragments like 123, 109, 95, 83, 76. So this is known as the mass spectrum for the compound eluted in the gas chromatography at 20.31 minutes. So this is what we get from the device. So we need to compare this spectrum to the spectrum taken under the same analytical condition in one of the very well-known and reliable libraries which are available for free online. For example, NIST chemistry workbook on the website shown on this slide here. You can show it later, and in your material also it's available for you. You can go for this website and you can select search and then you can search for citronellol, and this is what you get. Exactly the same molecular ion peak with the same fragment ions like this. This is the mass spectrum we got from NIST, but have to be analyzed under the same experimental conditions. So this is very important to use the technique or the online resources which can serve the question I'm going to ask. Here I'm trying to analyze very simply essential oil and analyze non-polar molecules, low molecular weight. So we used the very simple, very old mass spectrometry technique, which is called the mass electron impact mass spectrometry technique. Then we can go further to understand what is these fragments we got from the nest or from our analysis. What is this? 123 and 109 and 95 and 83 fragments. We have to do some efforts in chemistry, starting from our structure, citronellol here, containing 10 carbon atoms. It can lose a molecule of water to give another ion at 138, losing another methyl group to give another ion at 123, losing either propyl group or methylene group to give other ions at 95. So these ions, the ions which you have seen in the mass spectrum at 138, at 123, and at 95. We have to confirm the fragmentation pattern with our fragmentation scheme for the target compound. And we have to go like this for each compound in our sample. So a little bit, it takes time, but it can confirm and give us an accurate results and show us what are the compounds present in our sample. A very simple technique, but it has to be done in the right way. And as I said, this uses the very simple form of mass spectrometry, the very old technique, which is electron impact ionization or EI mass spectrometry. Then we will have another example. Here we analyze another plant called stevia, one of the very famous sugar substitutes. We use this plant, the leaves of this plant, either in powdered form, dried and powdered form, or we extract some compounds from this plant, from the leaves of this plant, and we use it as a natural sweetener for people like to don't have like to use sugars, low calorie sugar substitute, or for diabetic people. So the leaves of this plant containing compounds called steviosides, kind of diterpenoid compound containing 20 carbon atoms, also still low molecular weight compounds, still the non-polar compounds, and these compounds are derived from the leaves of the plant called Stevia ropediana, very important pharmaceutically, from pharmaceutical point of view. Here we try to make a tissue culture, another alternative or substitute for the field cultivated plant. We try to produce callus cells from the stevia plant using the principle of plant tissue culture, or زراعة الأنسجة النباتية. And then we made an extract from our plant and from the plant which is cultivated in tissue culture, and from the cells, the callus cells produced by some hormonal impact or hormonal composition on this plant. Then the question we want to answer here whether the steviocyte are produced in our cultured cells or our cultured plant. Okay. So this is the result we got. We made an LC mass analysis. We used the different technique, liquid chromatography, HPLC, coupled to mass spectrometry. In this case, we used the electrospray ionization technology for ionizing our molecules steviocyte, target molecule steviocyte. We got this LC mass chart for the field grown plant here. We can easily see, divide this chart. Some of these signals represent our target compounds, steviocyte, while the other signals represents the phenolics produced by this plant. Our target here is steviocyte in the cultured plant. This is the stevia plant, cultured in jars on some media, and this is the callus produced from this plant. Callus means undifferentiated tissues, doesn't show any organization or differentiation, like a bacterial colony, but coming from plant, not from bacteria. Here we analyze the 3 extracts obtained from the field-grown plant, from the cultured plant, and from the callus. We want to know by the LC mass technology. Mass can be very helpful here to identify what kind of compounds produce the callus, or whether the callus can be a source for the steviocyte, for these pharmaceutically important. Okay, very sorry, very quickly. So this is the. Just I finish quickly. I'm sorry, I'm speaking very slowly for everybody to be very simple. I'm sorry for this slow mode, but I'd like to be very. Yes. So we go for this. We got also the mass spectrum here for our compounds. For example, here compound at 8035695, and call it high resolution mass, can give us 4 digits, can identify the compound very accurately. We go for some of the databases, like called here a mass bank. Very many databases are available on the internet. You have to choose or have to select the one which is reliable for you, the one which is giving you accurate data. Here's the mass bank. We enter our data, and depending on using this table, we can identify which compounds, and we confirm our data from the mass bank. Thank you very much. I'm sorry for exceeding my time.
Thank you, Dr. Sama, for this lecture. I just want to add just very small comment, just a comment. Whenever you isolate compounds from, let's say, a natural source, and you suspect that your compound, let's say, is A. So Dr. Sama said, okay, we shoot it on the GC first, and you see the retention time. This is the first step. Then you analyze the mass spec, and you use the library just to make sure, 100% sure, if you have authentic samples, then you should shoot it on GCMS to confirm and to say that the retention time and mass spectral data agree with the authentic sample. Just, all right? Thank you. Dr. Sama. Okay, for training session, our next speaker is Dr. Fadwa Oda. Dr. Fadwa is my colleague at the Department of Chemistry. She got her bachelor's degree and master's degree from the University of Jordan. Then she went to the States and got her PhD from Clarkson University. Then she joined the faculty, our Department of Chemistry, 25?
A long time ago.
Yeah. And Dr. Fadwa is, I consider her like an expert in NMR. She did some work on our NMR machine. Then she supervised some work at Clarkson University. She got actually a scholarship there, right?
Yes.
Okay. And Fadwa has over 40 published research articles, review articles. She supervises and is supervising some MSc and PhD students and undergraduates too. And she's going to talk to us this afternoon or today about solid state nuclear magnetic resonance spectroscopy, supposedly a training, right, for students?
Yeah.
Okay.
Very simple. I'm
Floor
going
is
to give
yours.
it. Thank you very much.
You got 40 minutes?
Yes, you got 40 minutes. Thank you.
Sorry.
What? Okay. So I'm going to be fast. Usually I'm very fast in presentations. So yesterday, the day before yesterday, and today we have been talking about different spectroscopic techniques and characterization technique, NMR, mass spectrometry, and so on. All of the NMR discussed was about concerned with solution NMR. Okay. Here I'm fast. And of course, NMR can be used to study any nuclei that is magnetically active. So we can do.
Online people are complaining. Sorry.
Can I take this one? No, this one.
No, it's okay. Share screen is better.
Okay.
Yeah, that's what they're saying.
Is it working now?
Better, but you'll tell me.
Okay. Those who are on Zoom, please tell us if it's working. So we can do proton NMR, we can do deuterium NMR, fluorine NMR, any nuclei that is active, so we can do NMR for it. Now, in order to do NMR, we put our sample in a magnetic field, external magnetic field, so that the spins are aligned, and due to the difference in population and the resonance between the two states, we can obtain our signal. Of course, the difference in populations between the two spin states is very small. That's why we can do one of the three ways or all of them. Either we increase the spin or lower the temperature, or increase the magnetic field, which will increase the frequency, and then we will get higher population differences. So this has been discussed in previous lectures. Now, if I have a compound that I want to characterize, usually we depend on X-ray, single crystal, like the one was talking about Professor Betzel or Dr. Kras, who is going to talk this afternoon, or we go to solution NMR. But if you are going to use X-ray spectroscopy, you need a crystal. If you don't have a crystal, you cannot really work with X-ray. And if you want to use NMR, the one we are familiar with, which is solution NMR, you have to have your compound soluble in a suitable solvent, and this solvent should be deuterated in order to look, and of course, if you use normal solvent, they will saturate the whole proton spectrum of you. Also, you will have a concentration problem. Even if you use solvent that does not have protons, you will need to lock the instrument. Now, what if your samples are not crystalline? For example, you have a cement sample, or you have a soil sample, glass samples. These are not crystalline samples. And what if your sample is not soluble in a suitable solvent? So it is solid. You have a protein sample, or a polymer sample, or a zeolite sample that is not soluble. So you need to do what is called solid NMR. Actually, the first NMR was for the gaseous state. Then it was for the solution state. Now it is for the solid and semi-solid states. So solid NMR actually overcome these two obstacles. For non-crystalline structures, you can use solid NMR, and for insoluble compounds or the ones that you have in very small concentrations, because you are going to put your sample in a solvent, so the concentration is a factor. But we have a problem in solid state NMR. These are the advantages that I talked about before. You don't need to solubilize your sample. There's no dilution of your sample because it's going to be as a pure solid material that you are going to pack in the rotors. We don't need to lock our instrument, and there's no need for deuterated solvents. Now the problems come here. Sorry. It's not very clear. If you have a solution NMR, you will have very sharp peaks for your sample. This is for carbon-13 NMR because they are aligned in the magnetic field. But if you have the solid sample, now the spin of the sample in the solid state is dependent on its direction. Some of the samples, they will be observable, others, some of the spins, others will not be because they are static. They are not moving. In the solution, they can be move and align themselves in order to have these two population, and we will have a homogenization. But in the solid state, we don't have such a thing. So the solution was to move the sample as if it is moving in the solution state, and move it with an angle. This angle so that we can remove all the distributions that are already present in solution, but they are canceled out. So we move the solid sample in a high speed with a certain angle from the magnetic field. We call it the magic angle spinning, so that we can have homogenization of our samples. Now, how is this done? Usually, now Ms. Rola will show you the rotors that we are using. Yes, but please don't touch them. You just look at them because they are sensitive to the to the moist. Yeah. So we bring our sample, we bag the sample, the solid sample, without any treatment, and then we close it in these rotors. If you notice these rotors, we have different types of rotors. The first rotor I used to work with is this big one, and it needed actually a huge amount of solids. So if you have a small amount of your solid and we were unable to back it in this large rotor, we used to mix it with KBR like we do in IR spectroscopy, and this will dilute the sample. Then we have other samples. Now this small one, of course, it depends on the instrument you are using, and it depends on the spin frequency that you are going to spin your sample. I think this one we can back around 1 milligram. Here we used to need more than 50 or 60 milligrams. So it is advancing. Now, as you see, we need to mark the rotors in order for the instrument to measure how many round per minute we are having. The larger spin velocity you will have, the more sensitive and nice splitting signals you will get. Also, we need an air to go and push these rotors in order to spin, and we use these it's like wings, and we make air go through them so they will spin. So this is the rotor. Now, and this is the probe. This is the solid state NMR probe. As you see, we put our rotor in here, and you see there's an angle. This angle is 54.7 degrees from our applied magnetic field, and there are air inlets from here, so it's going to spin, and there is like a detector that will count how many times we have rounds per minute. So, and this is different from the solution NMR. Solution NMR, we put them in our NMR tubes. I think you are familiar with them, the NMR tubes. For the students, I think there are students there. Please show them the NMR tubes. Okay, so how we are doing this? We are mimicking the movement in solution. So we are spinning the rotor with the sample back inside the rotor on the magic angle inside our probe. Then we do the frequency, the radio frequency, and we do exactly as we do in solution NMR. The trick here is the spinning should be really when you have a solid state NMR, the beak is very, very broad. And the broader the beak, the larger you have to spin your sample. So the spinning rate should be almost as the half height or half width of your beak. That's why in solid state NMR, it is not very useful for proton or fluorine NMR. It's better to use it for carbon 13, nitrogen, phosphorus, silicon, because we can have very nice signals there. So there are different solid state NMR methods. The most important one and famous one, it is magic angle. Spinning, here we are rapidly spinning the sample at the magic angle, which is 54.7. And usually we can reach more than 100,000, but for special instruments. And these spin rates depends on the mass probes we are using. Now, if we compare these three spectrum for phosphorus 31, this one is the static that it's not moving. It's just a solid sample that is put in the NMR and it's not spinning. And the other one, we are spinning the sample around 3,000 round per minute hertz, and here we are spinning it less by 400. So the larger, the faster you spin your sample, the better spectrum you will get. I can see it here, but I don't know if you can see it there.
The background.
I think we should change the background. Yeah.
Just make it a simpler background. Sorry.
Change it for all.
Yeah.
Sorry for that.
I don't know if it will apply to all. Let me just try.
I think we can apply to all. Format background, yeah. Go to the design. Okay.
Go to the design.
Design.
And change the design all
Here.
Yeah. Okay. Undo.
okay.
Some of the
Design.
things I'm writing white.
Okay. The first one. maybe.
The first one.
You know the problem. Some of the
Here.
writings are in white.
Let's see this one.
Let's see this one. Okay.
Let me see.
The
Let's
design will
see
change
the-
the font to the black.
Let me see. Let us see.
Yeah.
Yeah, it's
The design will change everything to the.
not bad.
Superb.
Okay.
Okay.
Thank you. Yes, thank you. And I'm always claiming I don't see well. Okay, I did them with the glasses on, so now I can see them. Okay, so we have different method for solid state NMR. The first one is with the magic angle spinning. Here is another example. I will provide these slides in the material. Number of scans also is another factor. If you have more scans with spinnings, you will have a better spectrum. Even though we are using a large number of scans at static, we don't have a very clean spectrum with good splitting. Here I am using only 216 compared to 324 scans, and here I have a very good pattern for splitting of my sample. This is 10 SN119 NMR, and I'm using, not me, of course, the ones who did that this spectrum. They use two methods. They use cross polarization and magic angle spinning at the same time. So you can mix between two methods. So spinning is really good. Number of scans, if you increase them with spinning, it also will improve the NMR of your solid sample. The other thing that is really very important in solid state NMR is the chemical shifts. Now here are different silicon NMR signals. Here I have a silicon atom surrounding by 4 oxygen atoms. I think they are with aluminum. I think this is a zeolite sample. Here, the other one here, one of them is oxygen -1. So I have a shift. Here I have a shift from this environment. Here I have 2. Okay. I have 2 negative oxygen ions and 2 OAl. So each time I'm changing the environment around the silicon atom, I will get a chemical shift. And these are called Q4, Q3, Q2, and so on. So let's have this sample. This is aluminum 27, and we have different, I have different aluminum neighbors around the silicon atom that I'm studying. Here I have 1, 2, 3, and 4 environments. Now, if you start modifying the surface, you will see how you are modifying the surface. Are you increasing the negative charge and decreasing the aluminum in your sample or not? Each modification on the surface, and this is solid state. We didn't do any solubilization or anything because these are, I think, zeolite samples. Every modification you do on the environment of the silicon, it will be interpreted as a chemical shift in your NMR spectrum. Also, this is also for the silicon 29 NMR. For potassium silicate, this is a glass material. As you see, I have a Q1, Q2, Q3, and with more modification, I have Q4. Usually when you do your glasses, you change the parameters in order to see what you are, how you are modifying them. For example, you put them at 500 degrees for 4 hours, 5 hours, 6 hours in the oven, and you modify the whole structure and the crystalline structure of your sample. Now, this is also 2 samples of zeolite. This is the original zeolite material. We have 1 type of silicon. It is the Q4. And then this zeolite was modified. And the result of modification, as you see, now we have not only 1 environment of the silicon inside my tetrahedral silicon center in the zeolites. Now we have more than 1, 2, and 3 other silicon environment in the zeolites. Of course, you can use this for porous materials. If you have membrane, which is semi-solid material, and you can see if you have absorption, if you have modification, interactions on the surface. So it is very powerful. This is a cross polarization. This is one of the most important techniques in solid state NMR. In this technique, we are transferring the polarization of a very abundant nucleus, like for example, proton or fluorine, and we transfer the polarization of this one to a less abundant magnetic active nuclei, like for example, carbon-13. Now we can do it in a very short period of time because we are seeing, for example, carbon-13 through the protons, which are abundant. They are readily in high concentrations in my sample. Of course, this is the pulse program. That's it. But I'm very fast. Wow. Originally, I was supposed to talk about the DNB mass NMR because this is the one I'm going to work with. Usually, I don't work with solid state NMR in my research. I work with dynamic NMR and diffusion. But I thought because there are so many students, just to keep it to the basics will be more effective. But if you have any question, we are all here. We have experts here.
Thank you, Fadwa, for your lecture. And we don't have time for questions. If you have questions for Fadwa, you can meet her at the restaurant while eating. Thank you again, and we see you at 2 sharp.
Because I know that many of the foreign delegates would like to go to the Dead Sea. So we make the lunch half an hour earlier. And we're starting at 2 o'clock.
2 sharp.
We starting at 2 o'clock in 1 hour. Then at 3 you can. All right.
At the first thing, I will tell you what is crystal structure and how we determine the crystal structure. I took a movie from a broker website. It's 4 minutes and will tell you how we measure the X-ray diffraction. So now, single crystal X-ray diffraction. I hope you will enjoy watching this movie.
In a single crystal X-ray diffraction experiment, a crystal is placed in the center of the instrument. The crystal is constantly rotated in a beam of X-rays, and a 2-dimensional detector records the signals from the sample. From the position and the intensity of these signals, the 3-dimensional structure of the sample can be unambiguously determined. Many minerals such as diamond and salt, organic molecules such as sugar, and drugs such as salicylic acid appear as small crystals. In these crystals, atoms, ions, or molecules are arranged in a highly ordered structure, forming a crystal lattice that extends in all 3 dimensions. This crystal lattice acts as a diffraction rating for X-rays. It is at this scale that the story of single crystal X-ray diffraction begins. X-rays are high-energy light with a repeating period called a wavelength, or lambda. Since the wavelength of X-ray light is similar to the distance between atoms in a crystal, X-ray diffraction can be used to determine the distance between atoms in the crystal. To understand X-ray diffraction, we first have to look into wave properties called interference. Very similar to throwing 2 rocks into the water, the circular ripples that result from the 2 stones seem to pass through one another as they propagate out from where the stones enter. This is known as interference. If the waves are in alignment, the signal is amplified. This is called constructive interference. If the waves are out of alignment, the signal is destroyed. This is called destructive interference. When an X-ray encounters an atom, its energy is absorbed by the atom's electrons. The electrons occupy special energy states around an atom. Since the absorbed energy is not large enough to release the electron, the energy must be re-emitted in the form of a new X-ray, the same energy as the original. This process is called elastic scattering. In a crystal, the arrangement of atoms, ions, or molecules forms a lattice with well-defined repeating distances. When planes in these lattices are exposed to an X-ray beam, X-rays are scattered by this regular arrangement. Strong amplification of the emitted signal occurs at very specific angles where the scattered waves constructively interfere. This effect is called diffraction. The angle in scattered beams is called 2 theta. For constructive interference to occur, the scattered waves must be in alignment, meaning the second wave must travel a whole number of wavelengths. In this case, 1/2 of a wavelength is traveled on the incident side, and 1/2 of a wavelength is traveled on the scattered side, resulting in 1 additional wavelength. In the case of the next X-ray, 1 wavelength is traveled on both the incident and the scattered sides, resulting in 2 wavelengths. This reinforcement occurs throughout the crystal. The exact angle at which diffraction occurs will be determined from the red triangle. The angle at the top is theta, half the angle between the incident and scattered beams. The long side is the distance between the atomic lengths, and the short side is 1/2 of a wavelength. The relationship between the diffraction angle and the spacing between the atoms can be determined by applying the sine function. Rearranging this equation yields an equation commonly known as Bragg's law, named after Sir William Henry and William Lawrence Bragg, the father-sons who won the Nobel Prize in 1915 for their work analyzing crystal structures with X-ray diffraction. Today, the technique of single crystal X-ray diffraction is used to determine the three-dimensional structure of a wide variety of crystals. X-ray diffraction helps scientists to identify the products resulting from chemical reactions and experts in the mechanisms behind these reactions. X-ray diffraction is also applied to determine the structure of proteins, which play a key role in all organisms, including humans. For example, our ability to see light depends on a protein called rhodopsin, located in the retina of our eye. Single crystal X-ray diffraction helps to understand the mechanisms behind seeing. Detailed knowledge of protein structures is essential to develop life-saving pharmaceuticals. Single crystal X-ray diffraction provides important information to finally understand the interaction of new drug candidates with the proteins in our body. As scientists push their ability to engineer materials on an atomic level, single crystal X-ray diffraction becomes an increasingly important tool in the toolbox. Advances in equipment design have made single crystal X-ray diffraction easier to use and more powerful than ever.
Thank you for listening to the movie. Now, let me close this one. We don't need it anymore.
Yeah, no problem.
Now, when the crystals are grown, we mount, myself in my case, I mount them on a glass fiber using a glue or like crazy glue or whatever you have. And we put it on the goniometer head and we put it in the middle of the X-ray as you have seen in the movie we just have watched. The first step to determine the crystal structure, we have to determine the unit cell. There are 7 crystal systems. Each crystal is composed of repeating units. These repeating units, we call them unit cells. The highly symmetric one, or the most symmetric one, is the cubic, and the least symmetric one is the triclinic. In the triclinic, which is the building block of the crystal, in the triclinic system, A does not equal B, does not equal C. A, B, and I thought one. A does not equal B, does not equal C, also alpha does not equal beta, does not equal gamma, we end up with the triclinic system. In the monoclinic system, beta is 90. So it's monoclinic is the same as the triclinic. The only difference is that in the case of monoclinic, alpha equals gamma equals 90. In orthorhombic, all angles are 90s. In tetragonal, A equals B, but does not equal C. Alpha equals beta equals gamma and 90. Hexagonal, rhombohedral, or trigonal, and cubic. So the first step, we need to determine the unit cell. If you couldn't determine the unit cell, you will not be able to solve the structure. This is the most important thing. You have to determine the unit cell correctly. After we determine the unit cell, we should start data collection. After we collected the data, as you have seen in the movie, the intensity of the spots, we have to do what we call it data reduction. Data reduction include like absorption correction and polarization correction. This is usually done automatically by the machine, and I will not talk about it today because I do not have enough time. When I was thinking how can I tell our students in 35 minutes about single crystal X-ray diffraction, this is very difficult actually. Now, but when we reduce our data, we should get from the machine two files, the HKL file and the RIS file. Now I will go and show you how to solve the structure. I will go to example 1. Myself, I use Yes, there are many programs can be used to solve a crystal structure. Now we have the RIS file or INS file, and we have the HKL file. Let's assume this structure is unsolved. We go to solve using OLEX. Myself, I use OLEX. Here you have too many programs. Based on my knowledge and experience, ShellX XT is the best. But let's use the older version, what I call it the older version, which is ShellX XS. We hit and we, also here you can see the quality of your data. The rint is 4%, which is okay. If the rint is higher, this number is higher than 10%, most probably you will not be able to solve your structure. And here our data is complete up to 49 to 2 theta 49. Actually, this data collected 20 years ago on a diffractometer point detector, which is it's very hard, or it takes too long time to collect the whole data. While now we collect to larger angle because we are using like CCD cameras to collect the data. Now we hit solve. Of course, definitely when you select ShellX, you can use either direct methods or Patterson. Usually we use Patterson when we have heavy atoms. Now let's hit solve. Now what we get, this is the bromine atom, this is the iodine atom. As you can see, these peaks represent electron density. As you have seen in the movie, X-ray is diffracted by electrons. Actually, what we see electron density, but based on the number of electrons, we can tell if this electron density is due to carbon atom, nitrogen atom, or other atoms. You can see here, based on our knowledge, there is a 6-membered ring. These most probably are carbon. So we carbon. Now refine. As you see here, I should. Now the R factor. What is R factor? R factor is measures the agreement between the measured reflections and the calculated based on the model of the molecule we are building. So the R factor is 8.78. The best to be 0, but this is impossible to get R factor 0. This is impossible. But in the idealized case, if you have perfect data, which is impossible, you will get R equal 0, but this is impossible. This means the smaller the R value, the better the structure you have, and the better the data set you collect. But based on what we have done, we are refining the structure, assuming the electron density is as isotropically distributed. But indeed, electron density is not isotropically distributed, it is anisotropically, because vibration of the atom along the bond is harder than the bending. So the atom or the electron density will appear as a banana shape. A banana shape. This means we say like refine using an isotropic model of the electron density. Now, as you see, the R factor reduced to 5%. Now, which one of them is the nitrogen atom? As you see, the program accepts and the R value is good, even though we put the nitrogen carbon, because the difference between nitrogen and carbon is 1 electron. So in the crystallography, maybe it's possible to cheat. But how do we know? If a crystallographer look at this structure, he will say why the thermal ellipsoid of this atom is abnormally small compared to the others. This means this is the nitrogen. The rule, if we add more, like we have here 7 electrons. If we put 6 electrons, the thermal ellipsoid looks smaller. I will show you now, we will put a bigger atom. Now the thermal ellipsoid looks bigger. So when we put it nitrogen and we hit refine, now the thermal ellipsoid looks better. Now let's put a heavier atom here. Go sulfur, we will end up with non-positive definite, which I do not want. Let's put fluorine. Refine. What do you see? The thermal ellipsoid gets bigger. So let's go back to nitrogen. Refine. Now, where are the hydrogens? Where is the hydrogen atoms? Actually, as we have said, X-ray is diffracted by electrons and almost no electrons on hydrogens because the electron is involved in the bond. In some cases, if we have a good data, we may be able to locate hydrogens. As you see here, the hydrogen here, we can see it. This is a hydrogen. This is a hydrogen. And this is a hydrogen. So our data is good. We can locate hydrogen. But we usually say add hydrogen. When we say add hydrogen, the program will put hydrogens in the calculated positions and add constraints on them. What does this mean? And constraints in them? It does not allow the constraints will not allow hydrogen atoms to move. Also, it add constraints on the thermal motion. So we will hit add hydrogen. Now it seems here, if there is an alert extinction, it says solvent, water, sweat. This happens usually when we collect data using a big crystal. Actually, as I have said, this crystal was collected using a diffractometer, and the detector is point detector. To reduce the time, I had to use a big crystal. This means, well, how can we do this? Also, we need to change the weight, and we click XT and we refine one more time. Now our value is reduced to 3%. Now we asymmetric. Yes, I will talk about asymmetric unit. What is asymmetric unit? I talked about the unit cell. How many molecules in the unit cell, or how many asymmetric units? Sometimes the asymmetric unit is composed of one molecule, as in this case, and forgive me, this is ionic compound. I'm saying one molecule. Probably I should have said one cation and one anion. It's one building unit. But I will show you that the asymmetric unit can be half a molecule or two molecules. In this case, let's abuse the term molecule. This is not a molecule, but this is the basic unit. One cation and one anion. Also, you have to select. Also here, it says cell contents from the unit instruction and atom list do not agree. I forget to mention something. When we solve structure here, what should we tell the program? At least we should tell the program what are the type of the atoms. You don't need to give him the exact formula, molecular formula, or empirical formula. It is enough to give him, like, we have carbon, hydrogen, or whatever you have. And sometime if there is a mistake while you are collecting the data or solving the structure, you will know the synthetic people think they have this atom. Indeed, they do not have. But this, during the refinement of the structure, you will know this. Now, most of the people, let me go back how to do this. You can do it using pen and paper to know what is the exact composition, the molecular unit, the asymmetric unit, the molecular unit, and the content of the unit cell. But myself, I'm kind of lazy. I do it using another program called Platon. Platon, one of the most famous programs in crystallography. This is the Platon. I go select COMSTECH. I go to COMSTECH. We are talking about example 1. Pick the RIS file and go publish tables. Now tell you Z, the number of units is 4, and the moiety formula is C5, H5, Br, and I. We go here, we edit it by hand. We go here, Z is 4, we said C5. 5 multiplied 4, this means we have 20, hydrogen 20. Also H4, yes, it says H4. This means this has 4 by 4, this has to be 16. No, it's a 20. 5 hydrogen. Yes, C5, BRIN. BRIN. Yes, there is no fluorine. Now, okay. Refine. This alert will disappear. It disappeared. Now, many people mix between two terms, crystal structure and molecular structure. Definitely, most of synthetic people, when they wanted to know what is their or what is the molecular structure of their compound. But actually, as you know, bulky properties depend on the intermolecular forces, on the self-assembly. So that crystal structure is more than the molecular structure. Crystal structure tells us the molecular structure and the supramolecular structure. If you want to look at the supramolecular structure, you go. I use a different program, which is Mercury. It's the default for this file. Here is like you want to see the short contacts. Here there is a halogen bonding between the bromine atom and the iodide, and there is the hydrogen bond. So actually 3-bromopyridinium, the two cations dimerize to form a dimer, and you can see like the whole supramolecular structure. So the crystal structure is more than a molecular structure. You can see also the supramolecular structure. Now, in my next example, I will show you how to find if you have a pure enantiomer in the crystal or racemate. In a principle, X-ray diffraction is not designed to tell the absolute structure, but actually we use what we call it the anomalous dispersion to tell if the structure we have is like 2 enantiomers, racemate, or absolute. In organic chemistry, the 1,2 disubstitute. Oh, I should use, like, this is a cyclohexane. Cis 1,2 disubstituted cyclohexane is like at room temperature in gas or liquid, it is a racemate of 2 enantiomers. You cannot separate them because the energy barrier to convert the first enantiomer to the second enantiomer is very small. You see both of them. You cannot separate them. But in solid state, definitely we can. These 2 crystals crystallized from the same solution. They were crystallized by Dr. Hamdallah Al-Hudalli. He gave me the 2 crystals. When I looked at the sample under the microscope, I saw this is little bit lighter in color, and this is a plate, and this is like sword-shaped crystal. I have never thought like of racemization in cis 1,2 dicyclohexane. When I collected the 2 structures, I will show you. The first one, keep in mind, I will talk about the lighter one. When I picked this one, the plate one, this is example 2. Now the structure is solved, but I will talk about how do we know if we have pure enantiomer or racemate. Here it is. It crystallizes in non-centrosymmetric space group. And the asymmetric unit is composed of 1 molecule. This means this molecule, or this is a pure enantiomer. If we hit refine, the R factor is 5%. When, in order to tell information about the absolute structure, we have to look what we call it Hooft parameter and the flag parameter. These are 0 within the experimental error. Hooft and the flag. Let's invert the structure. The command in OLEG to convert the structure is N minus B. Enter. Now we inverted the structure. If we hit refine, look at this number at the R factor. If we hit refine, look at the R value has increased from 5. This means this structure is wrong. And also the program warning us the structure should be inverted using NINV minus F compound. This means this is 1 enantiomer. If we go to example 3, if you look, it crystallizes in the tetragonal I4 sub 1 over A. This space group, this is centrosymmetric. If we hit refine, the R factor is 4.6%. Let's invert this structure and see what will happen. Definitely nothing is gonna happen because this space group is centrosymmetric. M minus F now the R factor stays the same. It didn't change. This means we have the two enantiomers in the crystal. This means this crystal is a racemate. And when we published this work, nobody thought of this before us. Actually, we were the first to talk about cis 1,2-di substituted cyclohexane and the racemization in the solid state. Actually, Dr. Muhammad tells me I have to finish. I wanted to show you how to refine if we have mixed occupancy, because sometimes if we crystallizes sodium chloride and potassium chloride, definitely we will find in the same crystal sodium ion and potassium ion. And there is a way in crystallography we can find the ratio between sodium ions and potassium ions. But since I do not have enough time to talk about this, if you have any question about solving crystal structures, small molecules, I'm ready to answer, and thank you for listening.
Thank you for this lecture. Let me start one question, or we can postpone this panel discussion. Maybe.
There is no panel discussion. Actually, I thought I will take the whole hour. It's like I should finish because yeah, because I want to, like, yeah, I think I need to tell the students how to define mixed occupancy. We changed the schedule already. Yes, this is what I thought of. Now, we knew how to tell if what we have in the crystal is pure enantiomer or racemate. Yeah, this we call them polymorphs. And I think we were the first to talk about the reason of polymorphism is due to different enantiomers. All of the people, they talk about polymorphism, packing polymorphism, which is due to different packing of the same molecules. Like benzene has two polymorphs. In the first one, the rings are packed or stacked above each other in this way. The other polymorph in solid state of benzene, the first ring is like this. The second is like a sword above of it. And we were the first to talk about polymorphism due to different enantiomers. And it's by luck, by the way. It's not planned. Now, also, I will tell you another point. If when we pick a crystal, is this the only crystal in the sample? Because we may pick this crystal and we have other crystal, other polymorph, other compound. In order to do this, we have to make, or we have to measure the X-ray, the powder X-ray pattern, and we compare it with the calculator. using mercury, we can calculate the powder pattern. This is the powder pattern based on the single crystal structure. We go and measure it experimentally, as here, and we compare them. How do we know if our phase is pure or not? If all the peaks in the blue, which is the measured one, there is a corresponding peak in the calculated one, this means our phase is pure. But if there is a peak in the experimental pattern and there is no analogous or corresponding peak in the calculated one, this means our phase is not pure. So in order to tell, is this pure or not, you have to measure the single crystal and powder X-ray. And if you measure the both and you get the calculated powder pattern the same as the experimental one, this means definitely you don't need to do any other spectroscopic technique to elucidate your structure. This is enough. Like in our case, if I measured the powder diffraction of these two crystals, I will find like the powder due to this crystal and the powder due to this crystal, and we do not have pure phase. The last one I will show you is example number 5. This. In this example, the asymmetric is composed of half a molecule. If we want to see the whole molecule, we write grow. Here, so the asymmetric unit fuse is a half molecule. If you look here, we have the bromine atom and the chlorine atom in the same site. And it says the occupancy of a bromine atom 0.65. This means almost 66% is the bromine and 64% is chlorine. I will kill the chlorine, kill or delete or kill, delete, and work. The occupancy 1, refine. Look at the R value, it will increase now. It goes to 6%. And the thermal ellipsoid has increased also. Now I put one bromine atom. So what I should do, I should increase the number of peaks. Define. Oh, it is still considered free. It says negative 3. Should go. Actually, it says negative occupancy. What does this mean? This means there is a minus part of the atom, which is impossible. The minimum in a place 0. It can't be negative. So I should go. It's the bromine number 1, I think. Bromine. In let's solve the structure from the vector. Okay. So we hit. As I told you, the best one is to use is Schillick's. So you see, Schillick's XT is the best, but we know this is copper. Refine. Let's put here 3 variables, or let's refine it to 3. If you read here, it says we have 0.8 bromine atom. There is 0.8 bromine atom. This means it's mixed occupancy. Some of them are chlorine, some of them are a bromine. So the average electron density, it looks like 0.81 bromine atom. Now, I will call this chlorine. I will call this chlorine, and this, let's remember the name because I should edit it manually. Or let's put it free. Let's refine it free. But now. Now. It says here we have 0.1 chlorine atom and 0.78 bromine atom. How do we treat this issue? We define this is Cl4 and this is Br04. We should do this manually. We should define a new variable, 3 variable. Here we write like 0.8. This is a 3 variable. We put it 0.5, but we should go to Cl4 This is the second free variable. This is the first one, and this is the second free variable. We go to CL1 BR4. Yep. I'm just copying it. I don't want to write it by hand. We tell the program, use the second variable. So 2, 1, this means use the second variable. And for CL4, this is BR04, CL4, this, where is CL4? We write -2, 1. This means assume there is a 1 atom here. The free variable is for bromine and the rest for the chlorine. We just hit OK and we hit define. Let's go and see what is the value of the free variable. Sometimes we should define more than one time. Probably we should have selected this is as the chlorine, not this one. It says here link to variable 2. Now we keep it defining. And if we go back here, we have non-positive definite. We should, like there, we should read this. But before we should go to the output file. What is the free variable? It's 0.79. This means 0.79 of the locations in the crystal, 79%, is located by or is occupied by the bromine and the 21 by chlorine. This means we can do elemental analysis. 21% is chlorine and 79% is bromine. And this is what I wanted to tell you today. And thank you for listening. Behind every structure is a crystal. Remember this. No, I'm done. Actually, I deleted some of my slides. I want to tell the student how to check without using the diffractometer if the crystals are single or not using the polarized microscope. But when I did the rehearsal yesterday, oh, I need more time, so I deleted the slides.
Thank you very much. Thank you very much for sharing today, and thanks all the speakers. Actually, Professor Abdiya just told me that after your permission, she would like to have a copy of this lecture because it was a very good lecture for the students, and she'd like to share it with her students and with the COMSTECH at the COMSTECH site as well. So after your permission, she's so happy with the lecture as a teaching lecture for the students.
Inshallah.
So now coming to the end of our workshop, just unseated for suggestions, questions. We like to hear from you so that for the future workshops, we can improve what type of suggestions, what is your impression about this workshop? So whoever wants to start, if you have any suggestions, I'd like also to hear from the students, and I would like you to share with me to cheer the students and thank them for their dedication, for attending. It was the flavor, they gave a very nice flavor for this workshop, and it was meaningful because of your presence. So thank you a lot. Is there?
I like the lyrics that thank you so much. And my suggestion is to have every
Our best, actually, the plan is to have more than one workshop per year with COMSTECH. So this is initially the plan. We will see how things are. All depends on funding. Whenever we have funding, we can do workshops. So, any from the students, would you like to share your impression? في انطباع كان حتى لو حبيت تحكوا بالعربي ما عندنا أي مشكلة، احنا بنحب تسمعوا عن هذه الورشة إذا بتحبوا. انبسطتوا؟ كانت كويسة؟ Was it good? Was استفدتوا؟
Thank you all for this workshop. I think we learned a lot from all of you at Jena University. And I think we're thinking now of coming to this university to learn and do research. Thank you for hospitality. And we really learned a lot. It's not all about theories and studying. You know, I think how to start with research or how to start our research and research. Right? So thank you all. But like I have a small request, can you send us all slides as well, so we can look at them again and benefit from the great research that they have done. So,
maybe Ms. Kazima may share some of the slides, some of the things.
This could can be turn it the other side so that people can see.
In addition to that, all the lectures that they hopefully are recorded for you. So I'll definitely check with my IT team back at home and then we can share the recorded form.
Thank you, Kazima. Actually, Kazima and I, we were working day and night for the past few months with Athena.
Thank you so much, Dr. David. Jordan, and all the arrangements that you have made. It's really wonderful. The vegetarian food is amazing. It's our most favorite food. The environment, the campus is extremely beautiful. And most importantly, I have seen more than 90% of us. So, the girls are very big. So, mashallah, all students here, they are really intelligent, hardworking, focused, and I'm so impressed the way they are trying to learn and new things, and it's really amazing. You are the kids, you are our hope. You can take your nation move forward. And you all are welcome with us at experience and the COMSTECH as a program for scholarships. So we could get some scholarship to apply for this program, visit us in the many scholarships. And we have special scholarships for signing. So try to come and we have free facilities. So we are already. So we have to work together to take some role in this work. Thank you so much. Thank you. Very nice comments, and Lord, I think you're an example to be followed. Yeah.
The workshop was very well organized, and the hospitality was heartless, absolute heartless in everything. And I express my gratitude and thanks to you, the organizer, and all the other staff here also. And I think you will be organized every year. Many thanks.
Thank you very much. Dr. Fadwa, Dr. Jalal, Khadija, there are many, and maybe we have already mentioned them before. We have really very active young Dr. Firas, Dr. Muhammad, Dr. Musa. Actually, the work was from a team that brought hopefully success for this workshop. Dr. Muhammad, would you like to add something?
Definitely was very informative. Definitely was very beneficial. Students, faculty. One of the most important things that people get out of these workshops or conferences is you know new people. You know new people. You meet new faces. You see other people from other countries, other culture, which is really important. You listen to different voices, different accents. So this is different for me, at least. You know, it was really an honor for me, privileged. I was really happy to know people, to meet them, to listen, to see what they are doing. And this is part of the larger world. Now the world now is not like what you do in Jordan is confined to Jordan. Everybody knows about it now. Just like this. So this is an opportunity for the young generation to look for future. You want to do some, say, higher degrees, go abroad or something. I'm sure these guys will be more than happy to receive their that. For the older generation like me, it's an opportunity also for cooperation. You know, I've been collaborating with so many international scholars because of these meetings. So I'm sure whatever the outcome is, it definitely is a positive outcome. On behalf of the organizers, I would like to welcome, you know, I was really happy to see you in Jordan, and I would like to see you more times in Jordan. And forgive us if there are some shortcomings. I'm sure that among the rest of the organizers and all of us try to make your stay in Amman as pleasant as possible. And I hope we succeed in that. So once again, thank you for coming to Jordan. And we wish you a very pleasant, very safe trip, flight, swimming, whatever, back to your country. Thank you.
Dr. Solomon, yes.
Thank you very much. I'm extremely delighted to be here. Remember, our visa almost did not come, and I was really worried that we may not be here. But finally, we work hard to make actually got our visa to come here. And I was expecting very good hospitality. The Arabs are generally very hospitable people, and you exceeded my expectation. You put a lot of effort and resources to make sure this event is successful. At the end of the day, we are human, and besides, it's very serious, but we are human beings. This human relationship is the most important. The relationship, where the collaboration we are going for, all those things are the most important. And I got to meet so many wonderful people in Jordan, and most importantly, from the country I'm very passionate about. A lot of students from Palestine. I'm really very excited, and we'll continue to be friends. And I know in this current age, there are so many ways of getting in contact with one another. Let's continue to do that. So this will be the first of many relationship we have. Thank you so much.
Thank you a lot for your nice encouragement and for extending opening the hands for the coming generation, for collaboration, for having them in the labs, doing their research, doing their PhD. So I hope this will be a good opportunity to every one of you. May I ask Professor to say something?
Yeah, thank you for giving me the opportunity. I'll follow up. It was very nice to come here. Thank you very much for the hospitality. The program was also excellent for me. I learned new things, or updated my knowledge. And as you said today, I remember in the opening session, there was also mentioned of scientific international collaboration. The science is outdoors. So humanity is facing these days many situations, but scientists they should be open-minded and collaborate, considering religion, politics, just ignore this. And looking back today, there are so many options to go for an internship for 4 weeks, 6 weeks, 6 months, 2 years. There are a bunch of agencies, Europe, the Erasmus program, which is almost semi-international. There are some programs, Science Without Borders. We have our country has the German Academic Exchange Challenge, which is also here. There's a German Jordan University. Jordan was here, was Google. So this institution has also an office from the Goethe Institution. So you can visit in person or you zoom in and talk with people, and you find many opportunities. So also there are funding agencies, stipendia, and the double degrees coming in favor, I would say. There's a bunch of programs offering double degree for internship and degree master for PhD from 2 universities. And it's certainly complementary to learning techniques on both sides and is valuable for your future CV. Last thing, I think we discussed couple words also. We are recommending for teachers now not only to focus on your research topic. These days in your CV, you need also documents for soft skill and key skill courses. So time management, enterprises, scientific writing, and lots of opportunities either here or you join a webinar. And this helps also for your future career. If you apply in future for a job or postdoc also, they want to see this type of software key skill courses. I recommend maybe for one of the future workshops, you invite 1 or 2 teachers for software key skill courses. Thank you.
A very important point of having these soft skills. Now nobody can live without them. They should take some courses, or we should focus also on our future workshop on such soft workshops. Thank you for your nice comments. Thank you.
as a Palestinian, Ministry of Power Education represented by Shahz, Najah National University represented by me as head of the chemistry department, my students at the chemistry department. We are happy to thank all of you for your efforts and you give us the opportunity to participate in this nice workshop. And really, it was very nice workshop for me and my students who are coming there. And we hope to participate in our workshop in the future.
You're always most welcome to come to Amman, to visit us in Jordan, and bring as many students as you can, because this is actually very enriching for the workshop, and it makes the workshop actually meaningful. Yes.
Good evening for all. Actually, I would like to thank everyone who gave us this opportunity, this amazing opportunity. Also, I would like to thank our head of department to encourage his students to be here. Thank you,
For your comment, yes.
Okay, in the name of Najah University, I thank all of you, the professors, the doctors. You put a seed of hope in all of us. I see like you're our scientific model. So inshallah, maybe in the future we have some collaboration with you. Maybe you will work together someday. Thank you for having us.
And thank you all. You were all the time asking questions, contributing in the discussion, having you in this workshop was very nice. Thank you.
One thing was missing. I mean, they knew about Jordanian food, but I would love to see or to hear something about the German cuisine. Pakistan. Well, Pakistan cuisine I know about it, but this is just a joke.
So it was a great pleasure having you all here. It was, in a way, as always, it's not easy to organize any event, but looking at your eyes, looking at your energy, looking at that it was successful, it just makes us feel so satisfied and so happy. So thank you for this nice energy back to us. I would like to thank a special thank to all the online and actually on behalf of all online attendees, I would like to thank you, Dr. Solomon, because you were taking care of the online attendees. So this gift is on behalf of online attendees.
Well deserved.
Well deserved. Thank you a lot for your help. You were really amazing. Thank you a lot. And now maybe we'd like to distribute the certificate for my senior colleagues. I feel really very humbled to give you the participation certificate. So they will be distributed later by, because I will give them to Kazima, and I will ask my friend and colleague, my dear friend Atia, on behalf of Professor Rahman, just to give the certificate to our beloved students. يعطيك العافية. اوكي وين بتحب إسراء عوان.
إسراء القاضي، شادي موسى، نادين عباس، أحمد تاي، إيمان، عمير، علاء جليل، ياسمين عياش، رند حازم، عرين يوسف، ضحى يوسف، ماريا محمد، صبا كمال، آية فريس، ريم عودة، دكتور آلاء خريس، دكتور توبي سلمى مسامح، شيماء الصوالحة، إسراء الأفغاني، أيان سعيدات، هبة سليم، سيرين محمد، عمر خلف، دكتور أحلام خرباب، براءة مشاكبة، منال الوحش، خليل صالح. سيما خالد. شروق سامي. غيداء. عمر خالد. صعبيتك كبيرة يا عمر. والله عمر مش عارف. دانا يوسف. دعاء كنانة. هنا عماد. طارق يونس. إسراء ناير. فينان. ضحى عواد. ضحى عواد. مايا محمد. المثنى حراقشة. محمد علي. بروفيسور دكتور عاهد زيون. بروفيسور دكتور شاهر الزيون. مصعب عبد الله. مبارك للجميع، يعطيكم الف عافيه. بدنا لو سمحتوا بس الوفد الفلسطيني بدنا صوره جماعيه لكم بعد اذنكم. صوره جماعيه. يعني عادي يا دكتور.
Bye bye, Zoom participant. Bye bye. Thank you so much.